Hello everyone, good morning. I guess I'm clearly visible, audible. Can somebody write something in the chat box so I can know if I'm clearly visible, audible? Students are still joining. Very good morning to all of you. Hello Harsh, hello Praveen. Am I visible, audible? The slides are visible. Let me open the slides. So hello everyone. So everything is good to go. We will start in the next few seconds. You can see my slides also, I guess. Okay. So, welcome you all for today's session. We are conducting a microbiology rapid revision, right? Your concise course for your FMG exam, right? So, we have already covered the general microbiology. We have already covered the immunology. We have started bacteriology, but few bacteria are pending. So, first, I will finish the few bacteria which are pending here, and after that, I will move to the virology. So, today we will finish virology, right? And tomorrow is the last day. So tomorrow we are going to complete entire parasitology as well as mycology. So that is a complete. Today, along with the mycology, few clinical uh, scenarios like clinical microbiology sessions will also I will conduct today. So first, let me finish. This is the plan. Let me finish the pending bacterias. It will take 1 hour or 1.5 hours to complete the pending bacterias. After that, I will start with viro and clinical. Since the plan is good, can we start? So just a second. Give me a minute. So can we go with this plan? Okay, so let me start with the pending bacteria. Okay, we have started the bacteria. I guess those who have attended my session yesterday, they may be knowing this classification very well. Okay, we have already covered gram-positive cocci. We have already covered gram-negative cocci. We have already covered gram-positive bacilli. So we have covered 2 bacteria here. We have covered 2 bacteria here. We have covered 6 bacteria here. A, B, C, D, L, M. In the gram-negative bacilli, we have covered all of them except Enterobacteriaceae. So today, what is pending in the bacteriology is Enterobacteriaceae number 1 and the 4 miscellaneous bacteria. These are very small. So we first let finish the bigger portion Enterobacteriaceae. After that, hardly 5 minutes are required on the small bacteria. So I will come on the miscellaneous. So this is the portion which is pending in the bacteriology, right? I guess this is the only pending portion, Enterobacteriaceae and miscellaneous. Rest all are already covered yesterday. Those who have missed, they can watch the recording. Please watch the recording. So let me start with Enterobacteriaceae. Okay, let me tell you something. Enterobacteriaceae is not a bacteria. Enterobacteriaceae is not a bacteria. It's a family having many bacteria, nearly 13 bacteria. But I will teach you the 6 important among them. Okay, I will not teach you all. So first understand that Enterobacteriaceae is a family. What is the page number, people? You can search now. What is the page number? Can anyone tell? Hmm? What is the page number of Enterobacteriaceae? Can anyone tell the page number in the PDF so I can tell everyone? Yes, it's 63. So page number 63, Enterobacteriaceae. Okay. So let me finish Enterobacteriaceae. So as I told you, Enterobacteriaceae is not one bacteria. It is a family of the bacteria. Okay. So how many bacteria are there? These all bacteria belong to Enterobacteriaceae family. Can you see the names? These all belong to Enterobacteriaceae family. So what is the basis of the classification? What is the basis of the classification? We classify Enterobacteriaceae based on McConkey agar. In the family Enterobacteriaceae, there are 2 type of members. Lactose is a sugar. We all know based on the lactose fermentation, we classify them. Some ferment lactose, some do not ferment lactose. So we take McConkey agar. The lactose fermenter form the pink colonies and non-lactose fermenter form the pale or yellow colonies. So can you see the lactose fermenter, they form pink colonies. And can you see the non-lactose fermenter, they form pale or yellow colonies. Based on which we are dividing the members into 2 categories. The Enterobacteriaceae members. Who are the 2 members? Say ma'am, lactose fermenter LF and non-lactose fermenter NLF. The LF will form the pink colony on McConkey and the NLF will form the yellow or pale colony on McConkey. Say yes. This is the basis of classification of Enterobacteriaceae. So you can see we are having actually three categories. The first category, as usual, lactose fermented, they all form pink colony, as I told you. I will read the names. The second family is non-lactose fermented, they all form yellow colonies. I will read the names. What is the third family? Third family is a late lactose fermenter. What do you mean by late lactose? Initially it is yellow, but later on, gradually in 24 to 48 hours, it get converted to pink. So it is something yellow also, pink also. Initially it is yellow, but later on get converted to pink. That's why late lactose fermenter. They directly form pink, they directly form yellow, but initially they form yellow and then convert to pink. How many of you got it? There are three categories: lactose fermenter, non-lactose fermenter, late lactose fermenter. The lactose fermenter form the pink colony, the non-lactose fermenter form the yellow or pale colony, and the late lactose fermenter convert from yellow to pink in 24 hours. Say yes, give me a thumbs up. Now I will help you to learn the members in each category. So you get many questions on the classification. So pink colony is formed only by 4 members. Please learn the name of the 4 members. The mnemonic is EEKC. Make some mnemonic, means I don't have full form. EEKC. E. coli, Enterobacter, Klebsiella, Citrobacter. Dekho, E. coli, I will teach you E. coli. Enterobacter, Klebsiella, Citrobacter. So EEKC. E. coli, Enterobacter, Klebsiella, Citrobacter, all these 4 form pink colonies because they are lactose fermenter. Okay, learn only 4. Okay, and remaining all are non-lactose fermenter. Don't learn the name. Just learn remaining all will be non-lactose fermenter. It can be Salmonella, it can be Shigella, it can be Proteus, Morganella, Providentia, Yersinia. All the members form yellow colonies except one. There are 4 types of Shigella. One of the Shigella is Shigella soni. So Shigella soni is a late lactose fermenter. You can learn a mnemonic. Soni is always late. Soni is late. It is a late lactose fermenter. So can you tell me the classification? Everyone with me? Everyone with me? I'm teaching you the last bacteria which was pending yesterday. Enterobacteriaceae. Anna, I told you it's not one bacteria, it's a family. So first thing you should ask me, ma'am, what is the classification? You tell me how many members are there in the family. Okay. So I told you the basis of classification is McConkey agar. It's also a PYQ. What is the basis of classification of Enterobacteriaceae? The answer is McConkey. The answer is McConkey. What are the 3 categories? You see, ma'am, lactose fermenter, which form pink colony. You see, ma'am, non-lactose fermenter, which form yellow or pale colony. And you see, ma'am, late lactose fermenter. So it is the 3 categories in which we are dividing: LF, NLF, LLF. Now tell me the members in each category. What are the members? Here we have only 4 members: EEKC. So E. coli, Enterobacter, Klebsiella, and Citrobacter. In the last category, we have Shigella soni. Remaining all will fall in this category. Remaining all will fall in this category. Okay, so these are the 3 categories. This is the classification of the Enterobacteriaceae family based on McConkey agar. Now, among them, I'm going to teach you these 4 or maybe 2 more. These 4 are important. So you have to learn these 4. Among all the 13 members, these 4 are important. We will see the detail of E. coli very detailed. Yersinia, Shigella, and Salmonella. Okay, so this family will be done. So let me start with the E. coli. Can I start E. coli? All the 4 members belong here. Among them, who is pink and who is yellow? Only E is pink. Remaining all are yellow. So I mean to say the E. coli is lactose fermenter. Remaining all are non-lactose fermenter. Among the Shigella, Shigella soni is a late lactose fermenter. So you see the classification. So first, let me start with E. coli. Let me start with E. coli. So E. coli. First, understand the diagram of E. coli. In the E. coli, this is the bacteria. This is the bacteria E. coli. This is the cell wall of the bacteria. Okay, this is the cell wall. In the cell wall, there is an antigen known as O antigen. So O antigen is always present in the cell wall. O is somatic antigen. Somatic का जो O है ना, it's O. So somatic antigen है ये. It's O antigen present in the cell wall. Please learn. Now E. coli के ऊपर capsule नहीं है. E. coli don't have capsule. You know the 9 capsulated organism, उसमें E. coli नहीं आता. लेकिन capsule like slime layer होता है. It is not capsule, लेकिन capsule के जैसा है ना. Similar to the capsule, there is a slime layer. In that slime layer, we have another antigen. The name of another antigen is K. K है ना. You will say, ma'am, K क्या है? K for capsule. K for capsule. You will see, ma'am, the spelling of capsule start with C. I know that. But this is not English language. This is Greek. In Greek, the spelling of capsule start from K. So the word is derived from there only. Greek में capsule is like this. K for capsule. It's not a real capsule, but a slime layer. So we have studied two antigen till now. O is present in the cell wall. K is present in the capsule. Apart from that, there are flagella. These are flagella and these are fimbriae. And also flagella and fimbriae. So the antigen present on the fimbriae is F. F for fimbriae. The antigen which is present on flagella is H. So total 4 antigens are present. See, the same diagram I have given. Got it? Everyone got it? What are the 4 antigens? Can you tell me the 4 antigens? Say, ma'am, O present in the cell wall. K present in the capsule. H present in the flagella and F present in the fimbriae. You can see the 4 antigens here. Say yes. Everyone say yes. How many type of O is there? 174. Don't learn the number. There are many types. K, there are 100 type of K. H, there are 53 type of H. How many combinations you can make? You will say, ma'am, O1, K1, H1. You will say, ma'am, O1, K2, H2. I mean to say you can make thousands of combinations. So I mean to say E. coli is of thousands of type, millions of type. There is not one serotype. So final serotype is written like this. What is the number of O? What is the number of K? What is the number of H? There are many O, there are many K, there are many H. F के टाइप्स नहीं पता है ना. So based on which we do the classification of the E. coli. समझ में आई क्या बात? समझ में आई? हाँ. So I'm teaching you a bacteria known as E. coli. I told you there are 4 antigen. O in the cell wall, K in the capsule, H in the flagella. Or last one is not important. F for fimbriae. You can skip also. So this is of many type, many type, many type. And we write like O is to K is to H. है ना? We write the number. O क्या? O120, K30, whatever. So we write like this. So there are many types of E. coli. But to understand, you should understand what is O, what is K, what is H. These are the 3 antigen present on the structure. O on the cell wall, K on the capsule, H on the flagella. This is the basic structure of the E. coli. Now coming on diseases. E. coli causes what disease? E. coli causes 2 diseases, diarrhea and UTI. Urinary tract infection and diarrhea. E. coli causes 6 type of diarrhea. 6. How many type? 6. See, it's difficult, but I will make super simple for you. Believe me. Listen. There are 6 type of diarrhea caused by E. coli. All the 6 are diarrhea only. सब में patient को तो diarrhea होगा, है ना? Okay. सब में patient को तो diarrhea होगा. लेकिन the mechanism is different. So I will tell you 6 different mechanisms for diarrhea. You got my point? I am going to tell you 6 different mechanisms for diarrhea caused by E. coli. So what are the names? Have you heard these names previously? Enteropathogenic E. coli, EPEC. Enterotoxigenic E. coli, ETEC. Enteroinvasive E. coli, EIEC. Enterohemorrhagic E. coli, है ना, EHEC. Enteroaggressive E. coli, EAEC. And diffusely adherent. See, I'm writing again. So you have to pick the word. Entero क्या? Entero E. coli is everyone, है ना? Entero E. coli तो सभी हैं. They all are entero E. coli. है ना? The last one is diffusely adherent. है ना? So you have to pick the word. Entero क्या? Enteropathogenic E. coli. Say the full form. Say the full form. Enteropathogenic E. coli. The second is enterotoxigenic E. coli. T for toxigenic. ठीक है? The third one, enteropathogenic, enterotoxigenic. The third one is enteroinvasive E. coli. I for invasive. The fourth one, enterohemorrhagic E. coli. The fifth one is enteroaggressive E. coli. So that word which is different is important. And last one is diffusely adherent. Diffusely adherent E. coli. है ना? So you have to learn the 6 names first. पहले तो 6 नाम समझो. Don't learn. It will be given in the options. You don't have to mug up. You have to understand this word is important. That will make the difference in that. है ना? उस word में उसका meaning hidden है. The hidden meaning in this word only. ठीक है? They all cause one thing. They all cause only one disease, that is diarrhea. भाई ये सबके सब एक ही चीज कराएंगे, diarrhea. Okay? They all will cause the diarrhea. Okay. So let me tell you how they all cause the diarrhea. The full form is written in front of you. You can see P for pathogenic, है ना? T for toxigenic, ठीक है? I for invasive, H for hemorrhagic, ठीक है? A for aggressive and diffusely adherent. Okay, so first learn the full form. The first thing, है ना? You should know the full form. Now see, they all are causing diarrhea in one master diagram. I will show you the 6 mechanisms. This is human intestine. In one diagram, I'm going to show you all 6 mechanisms. Listen, the master diagram. So you know we have intestine. The intestine is surrounded by the cells, है ना? The enterocyte. You can see these are the enterocyte. This is human intestine. So how in the human intestine they all are causing the diarrhea? See, one by one I will tell you. and the meaning is hidden in that name itself, है ना, in one word. ठीक है? So let's start with the first one. Imagine I am the subject. I am eating something. I am eating something or I am drinking something which contain enteropathogenic E. coli. My food or my water contain enteropathogenic E. coli, so it is going inside in my intestine. In my intestine. Hannah, what does it do? How does it cause diarrhea? Please understand. See the diagram. Enteropathogenic. P for pathogenic. No. So what does it do? It destroys the villi, microvilli of the intestine. It do not go inside. This bacteria, can you see the red color bacteria? This red dot is the bacteria. It is not going inside the cell. No. It is not going. It is not secreting any toxin. है ना? Neither it is going, वो ना तो खुद जा रहा है ना toxin भेज रहा है. Neither it is going by itself, or nor it is sending its toxin. No. What does it do? Pathogenicity. P for pathogenic. What do you mean by pathogenicity? It is destroying the villi. Villi. What is the function of the villi? Elizabeth, Kartika, Suji. What is the function of the villi? Absorption. Villi are destroyed. No absorption. Diarrhea. So what is the mechanism of diarrhea? Destruction of villi. Once the villi are destroyed, absorption will not be there. It will lead to diarrhea. So how I remembered this? No? How I remember P for pathogenicity. I remember like this. P for pathogenicity. So it will cause pathogenicity. What do you mean by pathogenicity? It destroys villi. It destroys the villi. It's visible in the diagram. Say yes. Everyone say yes. Do you have any confusion? I will show you six mechanism. On every mechanism, give me a thumbs up if you got it. Or indicate. If you don't want to give thumbs up, write down that you got it. Okay. So this is the first mechanism I told you. Okay. The second mechanism is enterotoxigenic. Please understand. It's difficult now. Toxigenic thoda difficult hai. Please see. Naam mein hi hai. The name itself depict. Toxigenic will secrete the toxin. Toxigenic. Can you see the red color dot? This red dot is the bacteria. ETEC. The name of the bacteria is ETEC. Bacteria is not going inside the cell. No, bacteria is not going. It is secreting a toxin. The name of the toxin is, there are two type of toxin, STLT. Stable toxin, labile toxin. STLT. And how does they cause the diarrhea? Let me tell you on the next page. Listen. So this is the intestine of the human. Okay, this is a cell of the intestine. This is also a cell of the intestine. The human intestine. I'm drawing the cells of the intestine. Okay. So let me draw the enterotoxigenic E. coli, ETEC. Enterotoxigenic E. coli. This bacteria don't go inside the cell. No. It secretes toxin. The two toxins. What are the two toxins? Same name, LT and ST. What is the full form of LT and ST? Labile toxin, stable toxin. They are heat labile and heat stable. If you heat them, this one get destroyed. It's labile. If you heat it, it is stable. It is not destroyed. It is heat labile and heat stable. So these toxin will go inside the cell. Here LT is going, here ST is going. For example, I'm showing you two different cells. LT after going inside the cell, it will stimulate an enzyme. The name of the enzyme is adenyl cyclase. You know that. Adenyl cyclase. What does adenyl cyclase do? You tell me. It will convert ATP to cyclic AMP. So more and more cyclic AMP is formed here. Okay, what does ST do? ST will go inside. It is also stimulating an enzyme. But the name of enzyme is guanylyl cyclase. Guanylyl cyclase. समझ में आ रही है बात? Guanylyl cyclase. It also converts GTP to cyclic GMP. Cyclic GMP. So my point is that, please understand my point. LT increases cyclic AMP, and ST increases cyclic GMP. Make some mnemonic and learn. Don't confuse, L को A से जोड़ो, LT increasing AMP, और S को G से जोड़ो, ST is increasing GMP. Make some mnemonic. I don't have it, but you can make it, Local anesthesia, you can learn LA or SG, whatever you are. Please make some mnemonic. But LT, L for LT is increasing AMP. That is my point. And ST is increasing GMP. That you have to learn. Howsoever you learn, please learn. ठीक है जी, yes, that's it size, but you have to learn making some mnemonic. चलो मेरी बात सुनो, आगे कहानी सुनो. Continue the story. Listen, what does AMP or GMP do? AMP and GMP do the same thing. What does they do? There are sodium channels present. Here on the surface of the cell. AMP also block it, GMP also block it. AMP and GMP block the sodium channel. So sodium present in the lumen is not absorbed. No, because the sodium channels are blocked. Normally it is absorbed, but now it is not absorbed. Sodium will remain in the lumen only. Sodium के साथ साथ water भी. जहाँ sodium जाएगा वहीं water will follow the sodium. No sodium absorption, no water absorption. They are excreted in feces, in stool, causing diarrhea. So this is the mechanism of diarrhea. Say yes, people, say yes. Got it? Any confusion? So please learn. Harry, please learn. Please learn the two enzyme. LT stimulate adenyl cyclase and increase AMP. ST stimulate guanyl cyclase and increase GMP. So CMP and GMP do same. है ना? CMP and GMP, both of them block the sodium channel. So sodium channels are blocked. Sodium is not absorbed, water is not absorbed, leading to diarrhea. So this is the mechanism of diarrhea. Got it? Got it? Harry, got it? LT and ST? So LT is increasing AMP and ST is increasing GMP. That's what I'm saying. Because they are stimulating other enzymes. This is adenyl cyclase, this is guanyl cyclase. Everyone got it? Does anyone have any confusion? See, the same story is written. Everyone on the screen, everyone on your gadgets. See, ETEC. T का मतलब क्या है? Say the full form. ETEC. T का मतलब क्या है? What is T here? Toxigenic. T for toxigenic. Enterotoxigenic E. coli. है ना? Enterotoxigenic E. coli secrete two toxin ST and LT. है ना? So LT increases AMP and ST increases GMP, by which they are causing diarrhea. How they cause diarrhea, I told you. Okay, coming on the third one. Can I come on the third one? Say yes. Okay, can I come on the third one? Say yes. So we are done with the first, we are done with the second. So T for pathogenic, it is destroying the villi. T for toxigenic, it is secreting the two toxins, ST and LT. The third one is enteroinvasive. Invasive का Hindi meaning क्या है? What is the Hindi meaning of invasive? What do you mean by invasive? Invasive का मतलब क्या होता है? हाँ? Google meaning, dictionary meaning, invasive यानी घुसना, घुस के मारना, है ना? घुस के मारना. ठीक है? Listen. Invade inside, है ना? The pathogenic was not going inside. It was superficially destroying the villi. It was not going inside the cell. Please mind my words. The toxigenic also not going inside the cell. It was sending its toxin. It was not going itself. It was sending its toxin. But invasive, वो तो खुद जाएगा. बोलेगा, I will not send anyone. I will go myself and kill the cell. है ना? So ये घुस के मारने वालों में से. ठीक है? Got my point? है ना? So enteroinvasive, it itself go inside the cell. Can you see the red dot? It is itself going inside the cell and killing the cell. Right? The cell is getting ruptured. That is invasive. The name itself depicts invasive. Got my point? Now listen. When the cell will rupture, na, so here diarrhea to sab mein hoga. They all will cause diarrhea. But since the cell will rupture, it will bleed. So along with the diarrhea, it causes dysentery. It causes dysentery also. Got my point? It will cause dysentery also. Got my point? Dysentery means along with the loose motion, in the stool, bleeding is also there. So one of them causes bleeding only. That is enteroinvasive. The name itself depicts. वो घुस के मार रहा है ना, तो it will cause bleeding. The others will not cause bleeding. The others will cause only loose motion. But this along, so all cause diarrhea, but this cause dysentery. So you can understand that, है ना? This cause dysentery, ये याद करना है आपको. ठीक है? That is enteroinvasive. Coming on the fourth one. Can I come on the fourth one? The fourth one is enterohemorrhagic. H for hemorrhagic. Enterohemorrhagic E. coli. ठीक है? H for hemorrhagic. It is also secreting a toxin. The name of the toxin is virotoxin. What is the name of the toxin? Virotoxin. This toxin going inside. The bacteria is not going. The toxin, the virotoxin. Virotoxin का दूसरा नाम है Shiga-like toxin. लिख लो. Virotoxin और Shiga-like toxin एक ही बात है. The synonym. So it is secreting either Shiga-like toxin or virotoxin. It is going inside the cell and inhibit the ribosome. No ribosome, no protein synthesis. So cell will be destroyed. And in this way, it causes diarrhea. You got my point? याद आया क्या DPSS? How many of you remember DPSS? DPSS कल बताया था. याद है? हाँ? So diphtheria, pseudomonas, Shiga. What was last? Shiga-like. है ना? Shiga-like toxin. So it inhibit protein synthesis. है ना? It inhibit protein synthesis. So by inhibiting protein synthesis, it causes diarrhea. समझ में आई बात? है ना? Till now I taught you 4. Can you revise the 4? So shall I come on the 5th one? Anna, let me erase everything. Let me clarify everything for you. Let me erase and clarify everything for you. ठीक है, चलो. Enteropathogenic, enterotoxigenic, enteroinvasive, enterohemorrhagic. What we have studied? Enteropathogenic destroy villi superficially. It don't go inside. Enterotoxigenic secrete two toxin, LT, ST. LT will increase AMP and ST will increase GMP. And by blocking sodium channel, they cause diarrhea. Enteroinvasive is घुस के मारेगा. It will invade and rupture the cell. So it is not causing diarrhea. It causes dysentery. है ना? Enterohemorrhagic secrete a toxin that is virotoxin that inhibit the protein synthesis. That is the 4th one. Okay, coming on the 5th one, enteroaggressive. Enteroaggressive. It also secretes a toxin known as yeast toxin, but mechanism is unknown. We don't know yet how does it causes diarrhea. We know it causes diarrhea, but by what mechanism? We don't know that. I know. So it is not given in any book. You can skip. and the last one is diffusely adherent. The last one, say the name, last one ka naam bolo, diffusely adherent. Now you can see I have drawn the bacteria, all 6. You can see I have drawn a red dot, here also a red dot, here also a red dot, here also. But this one I have drawn something separate. है ना? It is having many fimbri on its surface, multiple fimbri. हर तरफ fimbri है. Pili or fimbri. What is pili or fimbri? The organ of adhesion. इसमें इतने ज्यादा pili or fimbri, it is having abundant of pili or fimbri, so it is diffusely adherent to the cell and making the cell inactive. So the cell will become inactive and it will cause the diarrhea. So that is the mechanism. So say people say, you got the mechanism of all of them? You got the mechanism of all of them? So this is the mechanism of all of them. In front of you, the mechanism of all of them. Now one important additional point in सबके बारे में. One additional point you have to learn for all of them. Okay, mechanism is clear. All 6. Basically, I taught you only 5. We don't know the mechanism of anteroaggressive. Rest all 5 I taught you. Okay, ji, है ना? Do you have any confusion? Now listen, one important point. Enteropathogenic, no additional point. है ना? No additional point is there. Just learn its mechanism. It is destroying the villi. That's it. है ना? No additional point. Enterotoxigenic, you know the mechanism STLT. Learn one additional point here, that it is the most common cause of traveler's diarrhea. Traveler's diarrhea. You know what is traveler's diarrhea? Whenever someone is traveling, the most common disease the person can have is diarrhea. That is traveler's diarrhea. Because person is eating outside food during the traveling. है ना? हम भी कहीं traveling करने जाते हैं तो sometimes we have the diarrhea, ना? So what is the most common? T for T. Traveler ka T, toxigenic ka T. Learn like this. T for T. You will never forget. It's a very common PYQ. So most common cause of traveler's diarrhea is ETEC. T for T. This is an additional point here you have to learn. This is the additional point. Okay? So EPEC, no additional point. ETEC, it is the most common cause of traveler's diarrhea. EIEC, invasive. Invasive में one additional point. It is the only one which causes dysentery. It do not cause diarrhea. It causes dysentery. है ना? Now can you tell me a test to confirm EIEC? EIEC का test बताओ. Imagine. Wait. I'm having a patient in my clinic. Today I'm having a patient in my clinic. Patient is coming to me and complaining, doctor, I'm having loose motion. I said, okay, what else? And the patient is saying, I'm having blood in my motion, not only loose motion. So basically the patient is complaining of dysentery. The patient is complaining of dysentery. So what sample I will collect? I will ask the patient to submit a stool sample. Now in this stool sample, I am suspecting, this is my suspicion, I am suspecting EIEC. I'm not sure whether it is EIEC, yes or no. I'm not sure. But I'm suspecting that. How should I become sure? How should I become sure? Very simple experiment. See, interesting experiment. Take a guinea pig. Take a guinea pig. It is an experimental animal. Take a guinea pig. है ना? This is the eye of the guinea pig. What you do now, you make saline suspension of the stool sample. Saline suspension of the stool sample and put one drop of the stool sample in the eye of the guinea pig. Yes, a dirty experiment. Put one drop in the eye of the guinea pig. I'm saying put the drop with the dropper. Don't inject it. है ना? Don't use needle. Use a dropper. With the dropper install. With the dropper install in one or both the eye. Whatever. है ना? Few drops of the stool sample of your patient. है ना? Now it is enteroinvasive. Yes or no, we want to know that. वही confirm करना है. I want to confirm whether enteroinvasive present or not present. So if it is invasive ना, it will invade everywhere. ये हर जगह घुसता है. ऐसा नहीं है सिर्फ intestine में घुसता है. It is not the thing it will invade only in intestine. It will invade everywhere. So here you have put it superficially. Now you have put superficially, so it will invade in the cornea and conjunctiva. It will invade in cornea and conjunctiva. It will invade in cornea and conjunctiva and causes keratitis and conjunctivitis. So if the guinea pig have keratitis and conjunctivitis after installation, the diagnosis is confirmed. And this test is known as Serini test. Nobody do so just for theoretical purpose. Learn what you have to learn. Learn क्या करना है? Learn करना है Serini test is for EIEC. Serini test is for EIEC. That's it. और कुछ learn नहीं करना है. Just learn. Learn the name of the test is Serini test. What is Serini test? I told you. So Serini test is for EIEC. That is an additional finding. So please learn one additional finding here. Serini test. Serini test. Okay. Can we move? So the next is enterohemorrhagic E. coli. Okay. The next is enterohemorrhagic E. coli. H. It causes 2 additional diseases. It causes diarrhea. They all 6 cause diarrhea. Diarrhea तो सभी करा रहे हैं. They all 6 cause diarrhea. But hemorrhagic cause 2 additional diseases. Number 1, HUS, hemorrhagic uremic syndrome. And number 2, hemorrhagic cystitis. So both are hemorrhagic. How to learn? H for H. Enterohemorrhagic is H. It causes H and H. Like T for T. है ना? Toxigenic is causing traveler's diarrhea. In the same way, H for H. In the same way, H for H. है ना? So hemorrhagic is causing HUS and hemorrhagic cholecystitis by some other mechanism. Here it is these two. For diarrhea, the virotoxin is there, but for these two diseases, it forms thrombus. It forms thrombus and cause these two diseases. Virotoxin do not cause HUS and hemorrhagic cystitis. EAEC, no other extra point you can learn, and here also no extra point. So few extra points are there. Give me a thumbs up, everyone got it? Now read. The 6, they are written in front of you. Have a look. Have a look. The first one is the enteropathogenic E. coli. The first one is enteropathogenic E. coli. What does it do? Just mark one word. It destroys microvilli. It destroys microvilli. That's it. That's it. That's it. है ना? Nothing extra. है ना? Enteropathogenic. एक ही क्वेश्चन आएगा इस पे. It destroy microvilli. और कोई क्वेश्चन नहीं आएगा. Only one question. The second is enterotoxigenic E. coli. So T for T. It is the most common cause of traveler's diarrhea. Please learn that. It secretes two toxins, ST, LT. You know what does LT do? Say, ma'am, it increases cyclic AMP. What does ST do? Say, ma'am, it increases cyclic GMP. And ultimately, cyclic AMP and GMP, by inhibiting sodium channels, they cause the diarrhea. है ना? So ST, so same is shown. है ना? You can see, don't learn, just understand. So you can see LT is going inside the cell, stimulating adenyl cyclase. Adenyl cyclase convert ATP to cyclic AMP. Cyclic AMP is inhibiting the sodium channel, causing diarrhea. Sodium is not reabsorbed. Water is not reabsorbed. Sodium and water in the lumen cause the diarrhea. So same thing is written. Okay. The LT, this is LT. Now ST, what does it do? It go inside the cell. It stimulates guanylyl cyclase. This was stimulating adenyl cyclase. This one is stimulating guanylyl cyclase. It converts GTP to cyclic GMP. Cyclic GMP also inhibits the sodium channel and it also causes the diarrhea. है ना? The next is enteroinvasive. नाम में ही है. Enteroinvasive. It invade. घुस के मारेगा. And not only diarrhea. Highlight the word. It causes dysentery. So in your question, whenever you are getting dysentery, you should think about it. So small clues in the question are very important. It is dysentery. For that, you do a Serenity test. Can you see the eye of the guinea pig? Anna, you have put the stool sample of the patient in the eye of the guinea pig. And after putting, you have put superficially with the dropper. It is going inside and causing keratitis and conjunctivitis. Say yes, people, say yes. This is Serenity test. Anna, you got it? Doxycycline, you got it? You asked me to repeat this now. So this is Serenity test. Okay. So we are done with this. We are done with this. We are done with this. The fourth one is antrohemorrhagic. Antrohemorrhagic secretes a toxin. The name of the toxin is virotoxin or Shiga-like toxin. एक ही बात है, Viro and Shiga. It inhibits protein synthesis. It inhibits protein synthesis. By inhibiting protein synthesis, by inhibiting ribosome, it causes diarrhea. It causes two additional diseases. H for H. 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 So it causes two additional diseases. What are the two? HUS, that is hemolytic uremic syndrome, and hemorrhagic colitis. So please learn the two additional disease. The mechanism is thrombus formation. The mechanism is thrombus formation. Now, there is a special strain. I forgot to tell you which causes these two diseases. These two diseases caused by O157H7. I told you there are million of types of E. coli based on O, K, and H. So learn one type. What is the type? Say, ma'am, O157 and H7. First learn it is a type of enterohemorrhagic E. coli. It is a type of EHEC. And I'm saying this is the most common strain which cause HUS and hemorrhagic colitis. इन दोनों का most common cause enterohemorrhagic है. I agree. लेकिन enterohemorrhagic का भी कौन सा वाला? I agree it is enterohemorrhagic, but which one enterohemorrhagic? There are millions of enterohemorrhagic, so it is this one. Which one? Which one? O157H7. You get a question like this, है ना? HUS is caused by, है ना? See the four options, है ना? I'm having four options: A, B, C, D, है ना? All four options contain O157H7. ठीक है? See, it's a PYQ. ठीक है? O157H7 of enteroinvasive E. coli, of enterohemorrhagic E. coli, of enteroaggressive E. coli, or enterotoxigenic E. coli. What is your answer now? You got my point? है ना? So first thing you must understand that HUS is caused by H for H. है ना? And it is the EHEC का type O157H7. Yes, in that case this is. So I want you to learn both things, है ना? HUS is caused by H for H, enterohemorrhagic, और उसका भी कौन सा type? It's O157H7. So answer will be B in that case. Yes, you all are right. So this is a confusing question. If you don't have the concept, you cannot solve that. Got my point? Okay. So this is the thing. You please highlight it and learn it. O157H7. ठीक है? Is it given in your notes? Antro-aggressive, although nothing important here, but learn. Have you heard about the HeLa and Hep2 cell line? Virology में आज पढ़ाऊंगी. Today I will teach you the cell line. You know, there are many cell lines. The viruses do not grow on agar plates. For that, we have to create the cell line. One of the cell line is Hep2. One of the cell line is Hep2. So take a Hep2 cell line. In the cell line, monolayer of cells is there. This is a cell line. है ना? It is Hep2 cell line. Hep2 cell line. So if you put the which one I'm teaching you? Antro-aggressive E. coli. One drop of EHEC here. So what does it do? All the cells एक के ऊपर एक खड़े हो जाएंगे. My point is that these all cells will be one above the other like this. है ना? Instead of monolayer, they will be like this. It is known as staked brick appearance. What it is known as? Staked brick appearance. है ना? हिंदी में कहूं तो ईंट का गट्ठा, एक के ऊपर एक bricks रखी हुई हैं. It look like that. The bricks are one above the other. है ना? So it is a way of diagnosis. EAEC. इसकी additional बात ये याद रखो. Can you see here? It is staked brick. है ना? These are Hep2 cell line. The cells are one above the other. Staked brick appearance. Please learn that. है ना? And last is diffusely adherent. It is having fimbriae or pili. It is having fimbriae or pili. That's it. So we are done. Say yes, everyone. Okay, you want me to summarize or it's done? Let's summarize quickly. Chalo. You will tell. Hannah, I will write the names. Chalo. The first one I taught you enteropathogenic E. coli. The second I taught you enterotoxigenic E. coli, enteroinvasive E. coli, enterohemorrhagic E. coli, enteroaggressive E. coli, and diffusely adherent E. coli. What I want from you all? I want the mechanism of action of each of them and one important point for each of them. First, tell me mechanism of action. See, ma'am, pathogenic में मुझे एक ही word याद आता है, microvilli. It destroy villi. It destroy villi, है ना? Toxigenic में मुझे toxin याद आता है. I remember toxin. See, ma'am, LT and ST. You know, LT is cyclic AMP, ST is cyclic GMP. We know that, है ना? Invasive, invasive क्या करेगा? Invade, घुसेगा, है ना? Hemorrhagic से मुझे VT याद आता है, virotoxin या फिर Shiga like toxin which inhibit the protein synthesis. ठीक है? Enteroaggressive mechanism is unknown. Make a question mark. We don't know. It's not given in the book. है ना? And diffusely adherent. It is having pili or fimbriae, whatever you say, by which it is causing the diarrhea. So this is the mechanism. Now I will tell you one important point in each of them. Please. Enteropathogenic, no important point. है ना? Enteropathogenic, no important point. Enterotoxigenic, T for traveler's diarrhea. It is the most common cause of traveler's diarrhea. है ना? You get question on that. Enteroinvasive में मुझे दो important point याद आते हैं. Number one, it is the only one which causes dysentery. Number one. And number two, Serini test. There is a test to confirm that. It is Serini test. So Serini test is for enteroinvasive. Please learn that. H, see there are two more H. H for H and H. So one is HUS, one is hemolytic colitis. है ना? So mechanism of both of them is thrombus formation. और इनका most common cause है O157H7. Please write that also. ठीक है ना? Aggressive में staked brick appearance. Staked brick appearance on Hep2 cell lines. And here, no important point. It is having the fimbriae. That's it. No important point. Give me a thumbs up. And you can't imagine how many questions are created from this one page. This is the summary table. You please learn. Learn the 6 names, the 6 type of the E. coli. Learn their mechanism. Learn their important points wherever applicable. Everyone. Do you have any confusion? Please ask. If you don't have the confusion, solve this question now. Solve this question. It's match the column. Match the column. And now match these with these. And now mechanism of actions are given. So tell me the mechanism of action of ETEC, EIEC, EHEC, EPC. Match. What is the correct answer from them? After matching, what is the correct answer? Can you please tell? What is the correct answer, please? Yes? Anyone? Yes, what is the correct answer? I'm waiting, people. Fastest finger first. Huh? So yes, Lalit, you are absolutely right. Yes, Prabingwal, you are absolutely right. Okay, you all are right. Okay, listen. So ETEC, T, it secrete toxin. T for toxin, है ना? So A का 1 ही है. Enteroinvasive, it invade. B का 2 ही है. Enterohemorrhagic, it form the thrombus. That is endothelial cell, है ना? C का 3 ही है. And pathogenic, it destroy the villi. So D का 4 ही है. Actually A1, B2, C3, D4. Actually there is no match the column, they are already matched, you can say. But you got my point. You can get a haphazard and you can do so for me. Now, if you don't have the knowledge, you can't solve this question. Do that, you cannot touch it. You cannot bluff. You cannot bluff. It's very difficult level question. It's not easy. It's not moderate one. It's very difficult level according to me. So if you have the knowledge, if you have the concept, then only you can solve it. Everyone got it? Why only few students answer? Others just sit. Huh? You got it, everyone? Okay. So can we proceed? We are done with E. coli. Here mainly you have to learn the 6 type of diarrhea. That's it. Now, coming on the next one. The next one is Yersinia. Let's come the next one. I'm teaching you Enterobacteriaceae family. We are done with E. coli and now I'm teaching you Yersinia. So let's start Yersinia. You remember bipolar या safety pin appearance? How many bacteria I told you? Can produce very high yield. What is Y? Yield का Y क्या है? Yield का Y क्या है? It is Yersinia. है ना? So we all know Yersinia pestis show bipolar या safety pin appearance. It is a bipolar or safety pin appearance. You all know that. Yesterday I taught you the stain is accumulating at the pole C. The stain is accumulating at the pole C and it is looking like the safety pin. Those who have attended my yesterday's session, they know what I'm talking about. You can see this is bipolar appearance. So bipolar appearance on which stain? Learn the name of the stain, either on Vason stain or methylene blue stain. So it gives this appearance. Learn the name of the stain, Vason या methylene blue. Okay, Yersinia से क्या याद आता है? Yersinia से मुझे एक ही चीज याद आती है, plague. It causes a disease known as plague. You know, plague के 3 pandemic आ चुके हैं वर्ल्ड में. There are 3 pandemic already occurred in the world of the plague in which millions of people have died in the past, long ago when we were not there. Our grandparents even not there, very long ago, है ना, hundreds of years ago. ठीक है, we have the pandemic, 3-time pandemic of the plague have arrived. It's a very deadly disease, है ना. So how does the plague occur? Again, I'm telling you, it's a zoonotic disease. So this is the 3rd zoonotic disease I'm teaching you. I taught you yesterday Bacillus anthrax. It's a zoonotic disease. I told you Brucella. It also a zoonotic disease. And now I'm teaching you Yersinia. It also a zoonotic disease. So कौन से animal की disease है ये? हाँ? कौन से animal की disease है ये? It is a disease of rodent. Rodent बोला तो चूहा. It is a disease of rodent. Yersinia. So natural host is rodent, not man. Not man. The natural host is rodent. It's a disease of rodents. है ना? Which rodent? जो हमारे घरों में होते हैं? No. It's a wild rodent. It occurs in the forest. Wild rodent. है ना? इसका नाम क्या है? Tiretta indica. The scientific name is Tiretta indica. The scientific name of this rat is Tiretta indica. Right? How does it occur to the human? How does it occur? Just suppose this is a rodent and this is a man. है ना? So how does it is transferred? The Yersinia is present in the blood of the rodent. है ना? So there is a vector. The name of the vector is an insect. It's like a small insect. Mosquito जैसा insect होता है. इसे कहते हैं rat flea. So rat flea take the blood from here and transmit it to here. And this is how it is transmitted from rodent to human via vector. The name of the vector is rat flea. So if I ask you who is the reservoir, so you will say rodent. And if I ask you who is the vector, you will say rat flea. Learn the 2 question. Learn the 2 question very quickly. You can see who is the reservoir. Say ma'am, rodent. And who is the vector? Say ma'am, rat flea. Tell me the scientific name of both. Rodent का scientific name है Tiretta indica. I told you Tiretta indica. Rat flea का scientific name है Xenopsella chiopsis. See, Xenopsella chiopsis is the rat flea. So this is rat, this is rat flea. This is Tiretta indica, this is Xenopsella chiopsis. How many of you got it? So here you get how many questions? You can see. They can ask you the reservoir, they can ask you the vector. Who's the reservoir? Who's the vector? Can you tell me who's the reservoir? Who's the reservoir? So rodent is the reservoir. Who's the vector? Rat flea is the vector. I know. So tell me the scientific name of both Tiretta indica and Xenopsella chiopsis. So please learn that. Okay? Please learn that. Okay? So this is the vector. And this is the cycle. There are 3 types of plague. You have to learn 3 types of plague. Bubonic plague, pneumonic plague, and septicemic plague. Let me explain the 3 types of the plague. How is bubonic plague occur? The bubonic plague occur by the bite of the rat flea. The rat flea is taking the blood. I know the same way. The rat flea is taking the blood from the rodent and biting human. So the point, just suppose it is biting here or biting anywhere. The point where it is biting, the lymph node of that point will get enlarged. And it is known as bubo. The bubo. Can you see here in the diagram? This is a bubo. See, this is a bubo. Bubo. What is a bubo? Bubo is a lymph node which is get enlarged. Where it is enlarged, not the complete body lymph node, only the point. Just suppose it is biting here. So that particular lymph node get enlarged and it is known as bubo. It is known as bubo. Got it? It is known as bubo. So this is bubonic plague. Please learn. Bubo से याद आना चाहिए, bubonic plague से याद आना चाहिए bubo. Everyone got it? Can we proceed? Just a second, है ना? I'm sorry. So yes, so it is bubo. Okay, the second is the pneumonic plague. The second plague is the pneumonic plague. What is pneumonic plague? Let me tell you what is pneumonic plague. Now this is the rodent. Imagine this is the rodent. How does it transmit? It is not via red flea. Imagine this rodent, and this rodent have plague. This rodent have plague, है ना? And the rodent is having pneumonic plague. The lungs are involved. So rodent also sneeze and cough. So the rodent is sneezing, है ना? And the rodent is coughing, and the human is in close contact of the rodent. So without red flea, it directly enters in the droplet form inside the lungs of the man. Say yes, you got my point, है ना? So human is in close contact with the rodent, not via red flea. Here red flea is not involved. Here red flea. So human is in close contact with the infected animal, and the animal is transmitting the infection to the human via droplet. Via droplet. Once it occurs to the human, from human to human, human to human, human to human, it will occur. Because this human will cough, give it to other. The other will cough, give it to other. The other will cough and give it to other. Like TB, it will be transmitted. So most of the transmission is from human to human. But first time it will come from rodent to human and then human to human. How many of you got it? How many of you got? This is pneumonic plague. But bubonic plague is not transmitted from human to human. The first one I taught you, bubonic. Bubonic is not transmitted from human to human. You got my point? Bubonic will occur to only to whom the red flea will bite. Red flea biting to this human, so this human will have bubonic plague. It cannot transmit it to other. Got my point? Bubonic plague is not transferred from human to human, but pneumonic plague transferred from human to human. It is through droplet. It is through droplet. है ना? Here respiratory symptoms are there. Respiratory symptoms are there. Does everybody got it? And the third and the last type of the plague is the septicemic plague. The third and the last plague is septicemic. What do you mean by septicemic? The first two plague, the bubonic plague and pneumonic plague. In bubonic plague, where is the Yersinia? It is present in bubo, that is lymph node. In pneumonic plague, where is the Yersinia? It is present in lungs. है ना? So here from buboes or here from lungs, when the Yersinia reaches the blood, Yersinia pestis reaches the blood, it is known as septicemic. So my point is that septicemic plague is always secondary. So bubonic convert into septicemic or pneumonic convert into septicemic. Septicemic is always secondary. Yes, septicemic is always secondary. How many of you got it? After going in the blood, it will cause gangrene of multiple organs. It can cause obstruction of the blood vessels. So that is the three type of the plague. We are done with Yersinia. We are done with Yersinia. I told you three type of plague. What are the three type of plague? Please tell me. What are the three type of plague? Please tell me. Say, ma'am, bubonic plague, pneumonic plague, है ना? Pneumonic plague and septicemic plague. So what is bubonic plague? It is the bite of red flea. है ना? Bite of red flea. How is pneumonic? Pneumonic is human to human via droplet. First time red flea से आया, but later on it is through droplet. And septicemic is always secondary. It is always secondary. Either bubonic or pneumonic can convert into septicemic. Do you have any confusion? Now read the question and tell me the answer. Read the question and tell me the answer. The three type of the plague. Now there is a farmer. Farmer is always a clue for you. You people read question and don't take the clue. भाई farmer बोला है मतलब farmer is in close contact with the animals. Farmer is in close contact. So they are talking about some zoonotic disease. The farmer word indicate that they are asking about some zoonotic disease. So whenever you eat farmer in your question, always think about the zoonotic disease. They indicate that they are talking about some that disease in which animals are involved. ठीक है? So farmer have fever. ठीक है? Painful lymphoadenopathy. Painful lymphoadenopathy. A farmer is having the lymphoadenopathy and fever. I know. So which stain will help in the diagnosis? Is it Nizar, Vason, Albert, or McFarlane? What is the correct answer? Vason stain. It will be Vason stain. The diagnosis is Yersinia, obviously. They are talking about lymphoadenopathy means he's having bubonic plague. They are talking about the bubonic plague. है ना? Bubonic plague है farmer को. So bubonic plague है तो Vason stain पे bipolar staining दिख जाएगी. So this is how you get the clinical question. How many of you got it? How many of you got it? Everyone? We are done with E. coli. We are done with Yersinia. Now I'm coming on the third one, Shigella. Very small, छोटा सा bacteria है. Shigella and finally Salmonella, so Enterobacteriaceae will also be done. And finally miscellaneous लेके we will finish bacteriology. And our next 1 hour, maybe 40 minutes. We will finish it. ठीक है, चलो फिर मुझे फटाफट वायरोलॉजी स्टार्ट करना है. वेटिंग eagerly to start virology quickly. ठीक है, I always want to start a day a fresh topic. But here it was pending, so I thought let's finish the pending soon, and after that quickly we can start with a fresh thing, virology. In one shot today I will complete entire virology. Okay, till evening. Today the class will be extended till the virology will be completed. Okay, so whatever, maybe 7, maybe 8, jab tak bhi hua, but I will complete entire virology today only because tomorrow is the last day, no? So tomorrow we will do parasite and myco. Okay, ji, okay, so be prepared. Okay, chalo. So let's continue with the Shigella. The third organism here is the Shigella. So let's start with the Shigella. What you know about the Shigella? There are 4 type of Shigella. है ना? Type A, B, C, D. The 4 type of Shigella. Shigella dysentery, Shigella flexneri, Shigella boydii, Shigella soni. Dysentery, flexneri, boydii, and soni. These are the 4 type of Shigella. Shigella dysentery, flexneri, boydii, soni. This is known as type A, type B, type C, type D. Soni के बारे में कुछ याद आ रहा है? कुछ याद आ रहा है Soni? Soni is late. I told you the mnemonic, no? So these 3 will form yellow colony. This also form yellow, लेकिन later on इसका मन बदल गया कि नहीं यार मैं तो pink बना लेता हूँ, है ना? So it converted from yellow to pink. The remaining do not convert. So my point is that the 3 of them are non-lactose fermenter. The last one is late lactose fermenter. Okay, 3 of them are non-lactose fermenter. The last one is late lactose fermenter. Okay जी, है ना? Please learn. So you get question on only this. Shigella पे यही question आता है. Repeated PYQ many times in your exam. Soni is late lactose fermenter. Please learn that by heart. Now I'm telling you the media, the culture media, है ना? Culture media of Shigella. Nutrient agar not important. They form translucent colony. Not important, you can skip. Blood agar पे भी gamma है. Not important. McConkey पे yellow है. No need to tell. है ना? They all form yellow except Sony, which will convert yellow to pink. They all form yellow. है ना? Transport media लगता है. They are very delicate, darling. याद करो transport media. So they require sex buffered glycerol saline. Please look your day 1 lecture. है ना? The general lecture where I told you the transport media. I told you 4 delicate darlings. One of them is this. ठीक है? Selective media. Learn 2 selective media. The first one is DCA. Don't learn the full form, have a look. Deoxycholate citrate agar. Deoxycholate citrate agar, DCA. On DCA, they form yellow colony. This is one selective. This is one selective. The other selective media is XLD. Again, don't learn the full form, have a look. Xyloslicin deoxycholate. Xyloslicin. Here they form red colony. So tell me the summary. Nutrient agar not important. If you want to learn, it's okay. Blood agar not important. है ना? If you want McConkey, yes, you have to learn McConkey. You have to learn transport and you have to learn selective. And now these 2 are not important. But McConkey, transport, and selective you have to learn. 2 selective media. Can you say blood agar is transparent, translucent? Nutrient agar. Blood agar is gamma hemolysis. McConkey will learn yellow, 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 except Sony, which convert to pink. है ना? Transport media is sex, buffered, glycerol, saline. ठीक है? Learn 2 selective media by heart. DCA, XLD. बोलो एक बार. DCA, XLD. DCA, XLD. Learn the full form if you can, otherwise it's okay. DCA yellow colony, XLD red colony. Learn the color of the colonies. So if image based question come na, you can identify. Imagine this image coming to you. They will give you the clue. It is not कि सिर्फ image दे देंगे. But you should be able कि the yellow colonies are seen in DCA. You should be able to find the red colonies are seen in XLD. ठीक है? Shigella also cause dysentery. Who else cause dysentery? अभी मैंने एक और बैक्टीरिया बताया जो dysentery कराता है. Who else? Currently I am telling you Shigella. Shigella cause dysentery. Dysentery मतलब blood and stool. One more I taught you na. Say ma'am, EIEC. One of the E. coli. EIEC causes dysentery. So like EIEC, जो घुस के मारता है, Shigella also cause dysentery. So what is the mechanism of action? What is the mechanism of action? Let me draw intestine. ये human का intestine है. Yes, है ना? So these are the cells of the intestine. Let me draw the cell of the intestine. These are the cells of the intestine you can see. Now there is a human. This human is eating the food which is contaminated with Shigella. So this is Shigella. So Shigella is present in the lumen of the intestine. The food and water, the contaminated food and water, it is going in the lumen of the intestine. This Shigella will do 2 things. Please understand the 2 mechanism very clearly. Ek to, the Shigella, number 1, it will go himself. It will go itself. It will go itself inside the cell and rupture like EIC. This mechanism is known as invasion. This mechanism is known as invasion, number 1. Number two, it will secrete a toxin known as Shiga toxin. And the toxin will go. It will not go. In some cells it will go, in some cells it will send its toxin. In some cell बोलेगा कि मैं खुद जाऊंगा, और कुछ में बोलेगा मैं अपना टॉक्सिन भेजूंगा. In some cell he is going himself, and in some cells he is sending its toxin. What is the mechanism of action of the toxin? You can see the red is the organism. This red color is the organism itself, Shigella, and the green color is the toxin of the Shigella, Shiga toxin. बताओ ना Shiga toxin का mechanism क्या है? ना बी इंटरैक्टिव यार. यू नो शीगा टॉक्सिन का मैकेनिज्म क्या है? इट इज जीपीएसएस. जीपीएसएस का वन ऑफ द एस है शीगा. इट इनहिबिट प्रोटीन सिंथेसिस. सो आफ्टर गोइंग इनसाइड द सेल, दिस विल bind to the ribosome, destroy the ribosome, destroy elongation factor 2. No ribosome, no protein synthesis, the cell will die. So by two mechanism it causes diarrhea. One is invasiveness, one is toxin. है ना? Which is more important? Which is more important for causing dysentery? So you will say, ma'am, invasiveness is more important for causing dysentery as compared to the Shiga toxin. Both are there, but if you get both the option, just suppose the question is like this. है ना? The question is like, what is mechanism of dysentery in Shigella? This is your question, है ना? And you get four options, maybe A, B, C, and D, है ना? The first option is invasiveness. The second option is toxin. You know toxin, which toxin? Shiga toxin. The third option is something. So my point is that if both are coming, na, you go with invasiveness. Although both are responsible, but this, if invasiveness is not given, go with this. You got my point? समझ में आ रही है क्या? हाँ? समझ में आ रही है? Maybe my voice is not very clear today. I'm having URTI, है ना? So that's why, but I'm trying my best. You are getting the concept, है ना? So virulence. The virulence factor are two. Please learn invasiveness and toxin. And please highlight invasiveness is more important than toxin. Invasiveness, both of them. So we are done with Shigella. Read the question. एक ही question आता है इस Shigella पे भर के. Late lactose fermenter. Who is late? Always late. Sony is always late. So Shigella Sony. I know Shigella Sony you have to learn. Okay. Coming on the last bacteria here, Salmonella. So Enterobacteriaceae will be done. Coming on the last one, Salmonella. Salmonella causes typhoid. I guess you all know it causes typhoid. It is a bacteria, the causative agent of the typhoid. So let's start Salmonella. Let me tell you the diagram of Salmonella. Here also we have the bacteria. This is Salmonella. Let me tell you the antigens. This is the cell wall. This is the cell wall. And this is the slime layer. Again, it's not a capsule. Salmonella is non-capsulated, but it's a slime layer like a capsule. भाई कैप्सूल नहीं है, लेकिन कैप्सूल की जैसी एक परत है, जिसे कहते हैं स्लाइम लेयर. सो अगेन O antigen like E. coli only. O antigen in the cell wall, है ना? There we have K. K for capsule. Here we don't have K. K is replaced by VI. We have K antigen in E. coli and we have VI antigen in Salmonella. That's it. That is the only difference. And the third, flagella. Flagella pe here also we have H antigen. I know we don't have fimbriae here. We have only flagella. So basically we are having three antigens. They are in front of you. What are the three antigens we are having in Salmonella? We are having O antigen, VI antigen and H antigen. ठीक है? O, VI, H. Now imagine this Salmonella enters in my blood. In any human blood. So does the body form antibodies against the three antigen? Yes. Human body will form antibody against anti-O also, anti-VI also, anti-H also. So my question to you, what is the order of antigenicity? Who is maximum antigenic and who is minimum antigenic? The order of antigenicity. Maximum antibodies against which and minimum antibodies against which? You know that. So the mnemonic is HOVI. Ho is maximum, VI is minimum. The mnemonic is HOVI. What is the mnemonic? The mnemonic is HOVI. So maximum antigenic is H and minimum antigenic is VI. Okay? So Lalit, I am saying this is a blood vessel of any human being. Maybe mine, maybe yours, any. Okay? Here the Salmonella is entering. You know the diagram of Salmonella, Lalit? So this is the Salmonella, you can see. So in the Salmonella, you can see this is the cell wall, this is the slime layer. So where is O antigen? You will see, ma'am, here is cell wall mein O antigen hai. Where is VI? The VI is present in the capsule. Capsule mein it is VI. And where is flagella? H antigen. The H antigen is present in the flagella, the third antigen. So we are having three antigen. Now in the blood vessel, when the Salmonella is present in the blood, so body will form antibodies against all. No? So human body will form anti-O also, anti-H also, anti-VI also. So three type of anti-O, anti-O bhi banega, anti-H bhi banega, because they all are foreign, and anti-VI bhi banega. समझ में आई? My point is that which antibody is maximum, which is minimum. I mean to say which antigen is maximum antigenic. Maximum antibodies किसके लिए बनेगी? हाँ, so the mnemonic I told you. The mnemonic I told you is HOVI. Ho के लिए maximum, H के लिए. The followed by O, followed by VI. So if you see the antibodies, these all are antibodies. You will say maximum antibodies are for O and minimum for VI. Got it? Quick confusion. So you have to learn the mnemonic HOVI. What is the mnemonic? Say it out. The mnemonic is HOVI. So make the marks. It's like this. So you will see now H is maximum antigenic, VI is minimum antigenic. Got my point? Have you heard about Vidal test? कितनों ने सुना है Vidal test? Everyone knows. Vidal test is for typhoid. So Vidal में we don't test the 3 antigen. We don't test. Just suppose I'm suspecting typhoid in me. I'm having fever. I'm suspecting मुझे typhoid है. You take my blood sample. Now there are 2 options. In my blood you can test the 3 antigen or you can test the 3 antibodies. है ना? So in Vidal we test antibody. We don't test antigen. We test antibody. Now see the order of immunogenicity. VI के लिए hardly any antibodies are formed. It's minimum antigenic. So we don't test antibody for VI. In Vidal, we test anti-H and anti-O. How much anti-H and how much anti-O? I will teach you Vidal also. I'm giving you a clue here. That is Vidal. I will teach you. Okay, got my point? है ना? Yes, yes, I'm coming on that, Medico. ठीक है? So let me tell you how does typhoid occur. Let me draw a human. Let me draw the GIT of this human. Let me draw the intestine of this human. Now, let me draw the blood vessel. Let me draw the gallbladder here, liver here, and let me draw the urinary bladder here. Okay? This person is eating the food which is contaminated with salmonella. The food contains the bacteria salmonella. The food contains the bacteria salmonella. So it will go inside. After going inside, it will produce ulcers in the intestine. The salmonella produce ulcers in the intestine. So ulcers, salmonella is typhoid. It will produce longitudinal ulcers. I will show you the diagram also. Longitudinal ulcers in the intestine. पहली बात ये याद करना है ना. After that, patient have high grade fever. Patient have fever, a typical fever, stepladder fever. Because of this, patient have fever. After that, patient have rash over the body. Red colored dots are there. I will show you the image. After that, the organism moves in the gallbladder. Okay, it remain in the gallbladder. It is disappeared from here. It remain in the gallbladder. It goes in the blood. Okay, gallbladder में से daily it is secreted a little bit. Whenever the gallbladder में से bile is coming, along with that it is coming. So every day a little bit it is coming. So that is a chronic carrier. Once it goes in the gallbladder, it is a carrier state. After that, it goes in the blood. In the blood, antibodies are formed. Anti-H, anti-O, anti-VI. VI is very less. We don't bother. Anti-H and anti-O. Got it? Anti-H and anti-O. Got it? So that is antibodies are formed. After that, from the blood, it goes to multiple organ. It goes to urinary bladder also. And it is excreted in urine also. Here it is excreted in feces also. So my point is that this is a patient of typhoid. My question to you, my question to all of you. This patient is coming to me. The patient is complaining, doctor, I'm having fever, I'm having pain, I have done the endoscopy, I can see the ulcers. So I'm having the suspicion the patient is having typhoid. What specimen would you collect? I'm giving you 3 options. Would you like to collect urine? Urine में आएगा organism. Would you like to collect stool? Stool में भी आएगा. Or would you like to collect blood sample? Blood sample में भी would you like to test the antigen or antibody? You have 4 options. One is urine, one is stool, one is blood ka antigen, one is blood ka antibody. Say 4 options. Say people say what sample and why? So you all are saying blood. Why blood? Why blood? Why blood? Why blood? So it is not blood. We take all 4 samples depending when the patient is presenting to us. So I will tell you. I will come on this point again. Wait. You can see the ulcers. Ulcers occurs in TB also. TB also, intestinal ulcers are common. Currently, I am teaching you typhoid. But the point is that in typhoid, the ulcers are longitudinal. See, what do you mean by longitudinal? And in TB, the ulcers are transverse. See, what do you mean by transverse? See, both are ulcers, but these are longitudinal, these are transverse. So how you will learn? TB में they are transverse and typhoid में they are longitudinal. Both are TT. कैसे learn करोगे? TB में they are transverse and typhoid में longitudinal. How I learn? Let me tell you. You know tie. Tie से ना मुझे tie याद आता है. हम tie लगाते हैं ना? We on a coat we put a tie. So tie is like this. कैसी होती है tie? The tie is like this. है ना? The tie. So tie is longitudinal. That's why typhoid से tie याद आएगा and you will learn it's longitudinal. So typhoid is always longitudinal. This is how I learn and never forget. है ना, ये मैंने 20 साल पहले याद किया, मुझे आज तक याद है, ठीक है. टाइफाइड इज ऑलवेज लॉन्गिट्यूडनल, टाई इज लॉन्गिट्यूडनल, है ना. और ट्यूबरक्लोसिस से ट्यूब याद कर सकते हो, द ट्यूब is like this, so it's something transverse, you can say it's transverse, so you can learn like that, you can make it out. Say yes, say yes. Got it? So this is how we learn. Anyways, here fever is also very peculiar. Fever is also very peculiar. I'm teaching you typhoid ka fever, है ना. Fever is also very peculiar. It's step ladder. देखो, day 1 patient will come say, doctor, I'm having fever, it's 99. Day 2 it's 99.5. Day 3 it's 100. Then 100.5. So gradually it is rising and not falling again. So this is known as step ladder, है ना. धीरे धीरे, gradually it is rising. Step ladder fever. Step ladder fever. Never forget. And rash is also there, especially on the back and the abdomen and the trunk. It is known as rose spot. So learn the ulcer, learn the rash, learn the step ladder fever. Step ladder fever is always indicating typhoid. है ना. Now I was telling you which specimen you will take. You will take all 4 specimen. All 4 specimen. है ना. Blood का antigen, blood की antibodies, stool and urine. है ना. The mnemonic is BASU. Let me tell you what is BASU. Let me tell you. Okay. So patient is coming to you. Imagine this patient is coming to me. ठीक है. Doctor, I'm having a fever. I'm having fever. So I will ask, have you done the charting? He will say, yes, the fever started 15 days back or maybe 20 days back. So initially it was very mild. It was hardly 98.9 or 99. Gradually it rises. Then I was having 99.5. Then after a few days it was 100. Now it was 100.5 and now I'm having 109. I know. So it's like the patient is giving a history of step ladder fever gradually. If the patient is doing the charting. If the patient is not doing the charting, it's okay. But yeah, if the patient is having rose spots also, है ना. So the day fever started, I will ask, tell me the exact day the first time fever occurred. First time, है ना. From that day, if the patient is coming within first week of the fever, है ना, I'm asking from the fever. From the fever, if the patient is coming within first week, take the organism is present in the blood. Look for blood antigen. है ना. If the patient is coming in the second week, na, organism gone from the blood, antibodies are there in the blood. Look for antibodies in the blood, that is Vidal. Look for antibodies in the blood. Both are blood. But here blood ka antigen, here blood ka antibody. In the first week, you get antigen in the blood. In the second week, the antigen is gone from the blood. It's antibody. It's antibody now. In the third week, blood में कुछ नहीं मिलेगा. Neither antigen nor antibody. Both are gone. If you want to make the diagnosis in the third week of the fever, go with stool sample. Mainly it will be present in the feces because it is present in the gallbladder. Every time it is secreted now. And in the fourth week, maximum yield is in urine. है ना. So the mnemonic is BASU. BASU. Learn like BASU. First week, second week, third week and fourth week. The sample you are taking, है ना. B से मेरा मतलब है blood का antigen, है ना. और ये भी blood है लेकिन antibody in the blood, है ना. S मतलब stool and U मतलब urine. How many of you got it? How many of you got it? So this is how you get the sample. You get many questions based on that. है ना. So I will not teach you all. I will teach you only one Vidal. How to perform Vidal. People say yes. Got it? You got it? What is Basu? है ना. You get a question like this. है ना. I have not put the questions today, so I want to frame it. Okay. Anyways, there is a 15. Let me frame a question for you. There is a 15-year-old girl, है ना, presented with step ladder fever. So first clue given to me is step ladder fever. है ना. पढ़ते से मेरे दिमाग में आ गया टाइफाइड, step ladder fever, है ना. She is having rash also, and she is having ulcers in the intestine, maybe longitudinal or only ulcers in the intestine. So I'm straightforward. My diagnosis is typhoid, है ना. The girl is coming to you on day 12, है ना. What sample would you like to say? The four options are A, B, C, D. The four options are: would you like to see blood antigen? Would you like to see blood antibody? Would you like to see stool sample? Or would you like to see urine sample? Yes, what do you want to say? At day 12, day 12 will come in second week. So second week answer will be blood की antibody or it's Vidal. It's Vidal. If I change it, day 12 को मैं कर दूं day 4, तो answer will be blood का antigen. और उसको change करके मैं कर दूं day 16, it will be third week, है ना. You got it. और उसको कर दूं day 21, तो हो जाएगा fourth week. So you have to see that. So accordingly, the question changes very frequently. You have to see day kaun sa. Based on that, answer will depend. Day kaun sa. And these are all your PYQs. Okay, ji. Can we go ahead? Anna? No, Vidal. I will teach you from the Basu. From the four things, I'm going to teach you only one. Vidal. A for antibody. Antibody matlab Vidal. I'm teaching you Vidal. How to perform a Vidal. How to perform Vidal. It's difficult. It's difficult. Anna? So I will try only once. ठीक है, समझ में आया तो अच्छी बात है, नहीं आया तो learn the summary, learn the conclusion, कोई बात नहीं. This is my patient. I am suspecting the patient is having typhoid. Based on the symptoms, the patient is having typhoid. I am suspecting that. ठीक है, what do? I want to take the blood sample, not blood. I want to take the serum of the patient, and I want to test the antibodies in the serum. I want to test the antibodies. है ना? Two type of antibody I want to test: anti-O and anti-H. Anti-H. I do not want to test anti-VI because I know the mnemonic HOVI. HOVI में H is maximum, O is there, but VI के लिए hardly any antibodies are formed. So I don't test VI. I test anti-H and anti-O. समझ में आई क्या बात? समझ में आई? Divide the patient sample into two. This is also patient sample. This is also my patient sample. The serum, है ना? But in one, I will test anti-O. I will do the testing of anti-O. In one of the sample, I will test anti-H. Both are same. I have divided the sample into two portions, half. है ना? This is my patient serum. This is my patient sample, patient serum. I want to know the antibody. So what I will do now? I will take 6 test tubes here, and I will take 6 test tubes here. है ना? Total 12 test tubes. ठीक है? 6 and 6. Listen. Commercially available O antigen is available in the market. Here I will put commercially available O antigen. It's available in the market. है ना? O antigen available in the market. ठीक है? So, and here I will put commercially available H antigen. So this is all O. This is all O. है ना? And this is all H. Fixed amount, same amount, maybe 1 milligram or something like that in all of them. I don't know the exact amount, but same amount in all of them. Say yes. So commercially available O and H are present. Now what you will do, you will take your patient ka sample and put it here. Serum, it's not blood, it's serum. So put it here 1:20, hai na? Now from here to here, half of it, 1:40, 1:80, 1:160, 1:240, and I guess the last is the control. hai na? The same you do here, here. The same proportion. Say yes, people, say yes. Now there are two options. Either the serum of the patient contain the antibody, so they will react with the antigen. Just suppose my patient is positive. So my patient is positive, my patient have anti-O also, my patient have anti-H also. So the anti-O will react with O antigen, the anti-H will react with H antigen. If my patient is negative, some alternate diagnosis, my patient have malaria or COVID or any other diagnosis because of which the fever is there, but typhoid nahi hai, so anti-O, anti-H will not be there. So they will not react here. That I want to know. Na? समझ में आ रही है बात? है ना? So here are 6 test tube, here also 6 test tube. Yesterday, have I taught you agglutination? Have I taught you the zone phenomenon? Prozone, zone of equivalence, and postzone. Have I taught you? If antibody is in access, prozone. If antigen is in access, postzone. समझ में आई क्या? So in these test tubes, the central few test tubes have zone of equivalence. है ना? यहाँ पे antibody is in access. यहाँ पे antigen is in access. So central 2-3 test tubes with zone of equivalence है. So there you will get the rings. And here also there you will get the rings. You will not get on first and last. You will not get on first and last because of prozone and postzone. So the last test tube where you get the ring here, the last test tube you got the ring here, that is your titer. See the diagram. See anti-H, anti-O. I want you to appreciate the 6 test tube. I want to appreciate 1:20, 1:40, 80, 160, 320 or maybe 640 तक लिया है. I'm sorry, 7 test tube लिया है ना? You can see this is H antigen in all of them, O antigen. You have to see ring कहाँ बन रही है. Where is the ring? You have to notice that is your titer. Say yes, that is your titer. So the cutoff is if O is more than 100 and if H is more than 200. O is more than 100 and H is more than 200, the diagnosis is positive. It's confirmed. I mean, it's a typhoid. It's typhoid. How you have interpreted? Say yes, everyone got it? Everyone got it? This is how we do the Vidal. Where was Vidal coming? I'm asking you. You have to answer. Let me give you a clue. Vidal कहाँ आ रहा था? मैंने कल पढ़ाया आपको. I taught you precipitation. I taught you agglutination. ठीक है? I taught you 5 type of precipitation. I taught you 4 type of agglutination. First, tell me Vidal was coming where? Precipitation की agglutination. पहला तो ये बताओ. It was precipitation का example और agglutination. Here it was ring, slide, tube, immunodiffusion and electroimmunodiffusion. याद आया? Ring, slide, tube. और वहां पे ring नहीं था, सीधे slide, tube, Coombs test, है ना? और last था passive. Yes, very good, Gokul, very good. So Gokul told us exactly it's tube agglutination. Agglutination तो है, लेकिन कौन सा? It's tube. Tube में I had given you 4 example. Yes, very good, Kartika. It was Vidal, V. Felix, Paul Bonnet. Yes. Very good. Hannah, so Vidal was there. So I'm just helping you to revise. Okay, so we are done. Read the question, tell me the answer. Okay, read the question. If you can write if some things are missing. Otherwise, I will share the annotated notes. Now, you can write whatever is missing. But hardly anything is missing. Most of the notes, 90% are complete. Hardly if some 10% are there, you can write now. Because every time we modify it. Every time. पहले मैं विडाल नहीं पढ़ाती थी, लेकिन एक बार क्वेश्चन आया तो I put it कि भाई विडाल भी पढ़ो. So every time we modify it. So little bit अगर थोड़ा बहुत रह जाता है तो you can write it. Otherwise, I will share the annotated every day. I guess you may be getting it now. Every day I'm sharing the annotated. Today also I will share. Chalo. What is the answer? There is a 15-year-old girl presented with splenomegaly, leukopenia, and fever. Died a few days ago. Longitudinal ulcers. सबसे बड़ा clue है. Long is the type. It's typhoid. है ना? If I change the question from longitudinal to transverse, answer will become TB. The same question will convert into TB. है ना? So we are done. So rest all the bacteria we have done yesterday. Okay. You want me to teach the miscellaneous also? I know. So I will share the miscellaneous. I'm teaching the miscellaneous. Hardly maybe another 20 minutes we are going to pack it. So let's start the miscellaneous. You have to give me a minute to jump on the miscellaneous. You also search. I'm starting with rickettsia. Where is rickettsia given in your notes? You search. And if it is not there, it's okay. I will share. Maybe usme nahi ho miscellaneous wale. I'm not sure miscellaneous is given in the notes or not given. I'm not sure. So look at the end of the bacteriology if it is given or not. If it is not given, I will share the PDF, or if you say Word file, I will share the Word file also. ठीक है? If you want to take the further print out, जो भी pending topic है, I will share a separate Word file. ठीक है जी? Give me a minute. Let me see where it is in my notes. Can you give me a minute? Just a minute. I want to open another PPT. Page number 75. People are saying you open, but maybe the complete notes are not there. I will share after the class. Don't worry. You give me a minute. I want to share another. I want to open another PPT. It will take 1 minute. Let me search my detailed PPT. Just a second, huh? Sorry for the trouble. Got it. Just sharing on the Telegram so that I can open it. So can you see my screen? Just confirm if you can see my screen. I have opened a new PPT. Can you see my screen? Can we continue? So I'm teaching you rickettsia. First of all, I'm teaching you rickettsia. I don't know whether it is given or not in the notes or not. I guess it is not given. Hannah, I will give you a time to write the summary. I will explain you. Hannah, in the next 10 minutes, I'm going to explain you complete rickettsia. Don't write even a single word. I will give you separate 5 minutes to make the table. And I will make a summary table. इसमें पूरा rickettsia cover हो जाएगा. I want you to make that table. If it is given in your notes, it's good. If it is not given, please make that table. I will give you in the end. I will give you in the end. Wait. Please understand first. Please understand. So let's start the chapter Rickettsia. है ना? In the Rickettsiaceae family, we have two bacteria to be covered, Rickettsia, Orientia. Please understand my point. Please don't write anything. Please understand first. समझना जरूरी है ना? लिखने के लिए तो मैं प्रोवाइड कर दूंगी. Don't worry. और लिखने के लिए मैं टाइम भी दूंगी. I will give you separate 2-3 minutes to write in the end. I will make the table ना मेरे साथ बना लेना. अभी मत बनाओ. देखो. So there are two bacteria you have to understand in this family, Rickettsiaceae में, है ना? One is Rickettsia, one is Orientia. There are 5 type of rickettsia. 1, 2, 3, 4, 5. I will teach you all 5. ठीक है जी? Orientia is only 1. So I will teach you 1. So total 6 organisms. I'm going to teach you 6 organisms now. Are you people with me? Let me open your Telegram group also. Where it is? Got it. So there are total 6 organisms. ठीक है जी? Total 6. I will show you one by one. So let's start first with rickettsia. The 5 type of rickettsia, and one is Orientia later on. So the 5 type of rickettsia. We divide the rickettsia into 2 groups, typhus and spotted fever. है ना? What do you mean by typhus? Here also patients have fever and rash. Here also patient have fever and rash. But here, along with the rash, patient have ischar also. Have I taught you what is ischar? Ischar का मतलब होता है कोल, कोयला, काला. So black color का ischar भी आता है, which was also present in Bacillus anthrax, malignant pustule. याद आया? So that is the additional finding here we have. Here we have two rickettsia. Here we have three rickettsia. So total 5 rickettsia. Total 5. I will show you one by one. Please understand. First learn the name of the 5. And at the initial look, if you are studying rickettsia for the first time, it may look difficult to you. If you have studied it already, it's okay. Otherwise, it may look difficult to you, but it is not that way difficult. Please believe. Anna. So let's start with the 5 names. So let me tell you the 2 typhus group. Typhus group में 2 rickettsia हैं. Write down Rickettsia prowazaki. Don't write, understand. RP. Rickettsia prowazaki is RP. Rickettsia typhi is RT. So learn RP and RT. Call them RP, RT. RP is prowazaki and RT is typhi. So please learn RP and RT. RP and RT. ठीक है? The second is the spotted fever group. The second is the spotted fever group. ठीक है? Please remove Afriki from here. We are not in Africa. We do not want to read the Afriki. Afriki is not important. Here we have 3 more rickettsia. RR, rickettsia rickettsii. है ना? Rickettsia conori. Kona होता है ना? Kona, conori. Rickettsia conori. है ना? RC. And rickettsia akar. Akar होता है ना? Shape. Akar. Akari. So there are total 5 rickettsia. Can you tell me the names? Names क्या है? सबसे पहले typhus group, spotted group. ठीक है? In the typhus we have 2. In the spotted we have 3. So just say the names. Rickettsia prowazaki, Rickettsia typhi, Rickettsia rickettsii, Rickettsia conori, and Rickettsia akari. Okay. So it is RTCRCA, PTRCA, PTRCA. Don't change the sequence. Make some mnemonic, but don't change the sequence. है ना? RP, RT, RR, RC, RA. है ना? You know the full form. Prowazaki, typhi, Rickettsia rickettsii, conori, akari. समझ में आई क्या? So first line, learn the 5 names, है ना? What are the 5 names? PTRCA. सबके आगे लगा दो Rickettsia, Rickettsia, Rickettsia. So say Rickettsia prowazaki, Rickettsia typhi, Rickettsia rickettsii, Rickettsia conori, Rickettsia akari. First learn. पहला step है 5 member के नाम याद करो. Orientia is only one, the 6th one. It's only one. The name is Orientia Susugamoshi. T silent. Don't say tissue. Say Susugamoshi. है ना? So the name is Rickettsia Orientia Susugamoshi. So total 6 organisms are there. The 5 Rickettsia and 1 Orientia. समझ में आई क्या? समझ में आई? So first learn this. Okay. The second thing you have to learn the disease caused by them. Learn the name of the disease. They all cause one disease. So learn the 6 diseases now. है ना? 5 or 6 diseases. So start with Rickettsia prowazaki. Start with the first one, Rickettsia prowazaki. Prowazaki causes a disease known as epidemic typhus. है ना? And Rickettsia typhi causes a disease known as endemic typhus. So both are typhus. How you will learn that prowazaki cause epidemic and typhi cause endemic? How you learn that? This is how I learned. देखो P for P. है ना? Prowazaki में P है और epidemic में भी P है. So P for P. This is how I learned that prowazaki cause epidemic typhus. Epidemic. And here I have learned ENT. है ना? Endemic का EN और typhi का T. ENT होता है ना? ENT. So this is how I learned. I never forget. है ना? So this is how I learned. ठीक है? जो epidemic typhus है ना? Sometimes it become latent for years and years. है ना? It become latent for years, and after that it become reactivated. So it is known as Brill's ulcer. So basically, Brill's ulcer is the latent version of epidemic only. It's not a separate disease. It's the latent version. Now coming on the remaining 3 diseases, spotted group. Spotted group में क्या diseases हैं? Please learn it once. Rickettsia rickettsii. It causes a disease Rocky Mountain Spotted Fever, RMS. Rocky Mountain Spotted Fever, RMS. Rickettsia rickettsii, RMS. Rocky Mountain Spotted Fever. ठीक है? Rickettsia R से R याद आएगा, है ना? RMS. Conori causes India tick typhus. How I learned? On the globe. Globe में India कहाँ है? Where is India? India is in a कोने में है. तो कोनोरी से मुझे याद आता है इंडिया, तो इंडियन टिक टाइफस. दिस इज हाउ आई लर्न. है ना? ठीक है. और आकारी, रिकेट्शिया आकारी, आकार से मुझे याद आता है शेप. है ना? और इट कॉजेज अ डिजीज नोन एज रिकेट्शियल पॉक्स. रिकेट्शियल पॉक्स को रिकेट्शियल पॉक्स क्यों कहते हैं? बिकॉज इट लुक्स लाइक स्मॉल पॉक्स. स्मॉल पॉक्स के जैसा दिखता है, नॉट अ स्मॉल पॉक्स बट इट लुक लाइक स्मॉल पॉक्स. दैट्स व्हाई. तो स्मॉल पॉक्स कौन कराता है? वायरस. और वायरस के कितने अच्छे शेप होते हैं. दे हैव द शेप. So I remember Rickettsia acari smallpox like rickettsial pox. Let me tell you how I learned. Listen, tell me the 5 bacteria. Say, ma'am, P, T, RCA, है ना? So say the full form, say the full form. These are the 5 bacteria. Now tell me the diseases caused by them. Who will tell me the diseases? Quick. Rickettsia prowazaki, P for P. It causes epidemic typhus. It causes epidemic typhus. Say yes, people, है ना? RT causes endemic typhus. It causes endemic typhus. We have learned like ENT. ENT, है ना? Okay. Epidemic typhus का ही latent version is Brills-Zinsser. ये मत भूलना. Brills-Zinsser भी वहीं पे लिख लो. ठीक है? R for R. Rickettsia, Rickettsia cause RMS. What is RMS? R for R. What is RMS? Rocky Mountain Spotted Fever. ठीक है? कोना, कोनोरी, कोने से क्या याद आता है? इंडिया. कोने से क्या याद आता है? इंडिया. इंडियन टिक टाइफस, ITT, है ना? और आकार, आकार से क्या याद आता है? वायरस. और वायरस क्या कराता है? पॉक्स. सो रिकेट्शियल पॉक्स. दिस इज हाउ आई लर्न, है ना? यू विल नेवर फॉरगेट, यू गेट मेनी क्वेश्चंस ऑन दैट. ये दूसरी चीज हुई. थर्ड और लास्ट चीज you have to learn here, the third and the last cheese you have to learn here. Okay, just a second. Is the vector. इन सब का vector बताओ. कौन सा किससे ट्रांसमिट होता है? They all are transmitted via vector. I will show you the images also. पहले समझ लो, है ना? So Rickettsia prowazaki is transmitted by louse. Louse को हिंदी में कहते हैं जूं. बालों में होती है जूं. Have you heard about the head louse? Louse, है ना? Body louse भी होती है. That is जूं, है ना? Epidemic, endemic typhus is transmitted by flea. It is transmitted by flea, है ना? RMS is transmitted by tick. Can you see? RMS is transmitted by tick. Indian tick typhus के तो नाम में ही है tick. So this is also transmitted by tick, है ना? And rickettsial pox is transmitted by mite. I will write, है ना? So let me explain you all 5. Then I will come on Orientia. देखो, सबसे पहला देखो, everyone on the screen, quick. Epidemic typhus. Epidemic typhus किससे होता है? Rickettsia prowazaki. Prowazaki, P for P, causes epidemic typhus. Who is the vector? Say louse. Louse, body louse. Learn the scientific name of louse. It is Pediculus corporus. Pediculus corporus, Pediculus humanus corporus. कभी-कभी scientific name आ जाता है. This is the diagram of the louse. This is the diagram of the louse, है ना? Clinical features सब में rash होगा, लेकिन learn the peculiarity of the rash in each of them. Here rash is everywhere except palm and soles. Except face, palm and soles. No rash on the palms, no rash on the soles and face. I'm talking about the first one. Rash is everywhere except palm and soles. Brills-Zinsser उसी का dormant form है, उसी का latent form है ना? Nothing extra. The second is endemic typhus. Endemic typhus is caused by typhi, Rickettsia typhi. Who's the vector? Say flea. Ma'am, flea. What's the scientific name of flea? Say Xenophila chiopsis. ये हम पढ़ के आ रहे हैं plague में भी. Plague में भी तो यही flea था. See the diagram. This is endemic typhus. The rash is same. Especially it is on the trunk. The rash is on the trunk, है ना? The third one is Rocky Mountain Spotted Fever. It is caused by Rickettsia rickettsii. The vector is the tick. This is the tick. See the diagram? This is the tick, है ना? The main thing is that here rash is there. ठीक है वो तो. It is most fatal. इन सब में से, इन पांचों में से highest mortality इसी की है. Most fatal यही है. Highest mortality and most fatal, है ना? And rash is maculopapular. The rash is maculopapular. I will make the table. Don't worry, है ना? The fourth one is Indian tick typhus. India, कोना. कोने से याद आता है इंडिया. इंडिया एक कोने में है ना? ग्लोब पे. So Indian tick typhus. Here also it's tick borne. Tick से tick याद आएगा, है ना? And clinical feature here is a ischar. The main thing here is a ischar, the black ischar. And the fifth one is the rickettsial pox. Pox, आकार यानी virus, है ना? It is caused by mite. It is caused by mite. Look at the diagram. Here also rash have ischar. Here also rash have ischar. That is not important. Here rash is vesicular. Vesicular यानी समझता है? It's like smallpox. इसके अंदर fluid है. If you rupture, the fluid will come. Here the rash is vesicular. You got my point? Huh? So in epidemic typhus, rash is everywhere except palm and sole. In endemic typhus, rash is on the trunk followed by extremity. In RMS, the rash is maculopapular. And in rickettsial pox, it's vesicular. Say yes, say yes. I will make the table, but समझ में तो आता जाए. Then you have to ultimately learn the table. सिर्फ table learn करना है. Vectors हमने सबके देख लिए. ठीक है? Say yes. Say yes. Okay. Mite and tick. Kavya, these are two different insects. जैसे mosquito होता है. Mite is different, tick is different. What is this question? This is mite. This is tick. These are two different insect, no? Insect ही अलग-अलग हैं. ठीक है? So the different vectors are there. Okay, got my point. So that is the thing. Last one, Orientia susugamushi. वो भी दिखा दो. Just a second. Questions we will solve. है ना? Let me show you last one, Orientia susugamushi. So we will be done. So we are done with the 5 Rickettsia. We will solve the questions. Wait. Last one is Orientia susugamushi. Orientia susugamushi. Orientia Susugamushi causes scrub typhus. The name of the disease is scrub typhus. Orientia Susugamushi. It causes scrub typhus. The name of the disease is scrub typhus. And here the vector is not mite, the bacha, the larva of the mite. The larva of the mite is known as chigar. The larva of the mite, not adult mite. The larva of the mite is known as chigar, chigarosis. And here also ischar is there. Here also ischar is there. Shall I make the table for you? Treatment for all of them is doxycycline. I will write this also in the table. ठीक है ना? Shall I make the table? है ना? Shall I make the table? Last thing मैं एक बताऊंगी wheel phalanx. बता देती हूँ फिर टेबल बना लेते हैं है ना. Last thing I'm telling you, the test is wheel phalanx. Now I taught you the 6 diseases है ना. What are the 6 diseases? Rickettsia prowazaki, Rickettsia typhi, Rickettsia rickettsii, Rickettsia konori, Rickettsia akari, Orientia susugamushi. These are the 6 I want to. These are the 6 bacteria. Then you have to write the diseases. Then you have to write the vector. Then you have to write the rash. And then how to make the differences between them. How to differentiate. And now, differentiate करने के लिए there is one test. This is known as wheel phalanx test. Wheel phalanx test. What is the difference? It is wheel phalanx test. ठीक है? It is wheel phalanx test. ठीक है? Let me tell you what is wheel phalanx test. This is a diagram of Rickettsia bacteria. On the Rickettsia, 3 antigens are present. What are the 3 antigens? It can be OK19, OX19, OX2, and OXK. है ना? Let me check. Yes, OX19, OX2, and OXK. है ना? तो किसी में कौन सा, किसी में कौन सा, किसी में कौन सा, that I will tell you. So please make the table with me, or I will give you separate time. Let me make the table. You help me in making the table. Tell me the name of the 6 bacteria first. Number one, you have to tell me the name of the bacteria. Tell me the name of the diseases they cause. ठीक है? Diseases के नाम बताओ. Tell me the name of the peculiarity of the rash in each of them. Rash तो सब में होगा, लेकिन tell me the peculiarity of the rash in each of them. ठीक है? Tell me the rash की peculiarity बाद में बताना. Tell me the vector in each of them. And after that, tell me the peculiarity of rash in each of them. And lastly, tell me wheel phalanx. So complete table on one page. I want you to make this table with me. You can take out your copy and pen and make it. है ना? So listen. Tell me the name of the 6 bacteria. So the 6 bacteria is PTRCA. है ना? So the mnemonic Mahima is giving PT Usha run. है ना? PT Usha run to Calcutta. कलकत्ता का CA. Good one. है ना? So you can learn. ठीक है? PTR का टुसु. ठीक है? और last one you can add Orientia also. These all are Rickettsia. The last one is Orientia susugamushi. Okay, PTRCA and susugamushi. Please learn. इसी sequence में learn करो. Don't change the sequence. Make some mnemonic. Do you have some or other mnemonic? And this one is also good. You can learn PT Usha run. कलकत्ता है ना? कलकत्ता और cat learn कर सकते हो. PT Usha CAT cat. You can learn something. जो भी learn करो, make some mnemonic. Don't change the sequence. I want that. Don't change the sequence. Are you people still with me? Live, alive, listening to me? Chalo. Tell me the disease caused by them. Who will tell me the diseases? फटाफट बताओ diseases. Rickettsia prowazaki cause a disease known as epidemic typhus. है ना? और उसका latent version is Brill's answer. यहीं पे लिख लो. ठीक है? Rickettsia typhi cause a disease which is known as endemic typhus. है ना? Rickettsia rickettsii causes RMS. है ना? It is causing RMS, Rocky Mountain Spotted. Rickettsia konori causes Indian tick typhus. ठीक है? Akari causes rickettsial pox. And Susugamushi causes scrub typhus. So total 3 typhus. देखो, epidemic typhus, endemic typhus, and scrub typhus. Please learn the name of the diseases. Howsoever you do. I told you the ways to learn. है ना? कोने से मुझे इंडिया याद आता है. आकार से मुझे पॉक्स याद आता है ना? This is how I learned. R से R. P से P. Please learn. Tell me the vectors. Vectors in each of them. Who will tell me? हाँ, यहाँ पे vector है louse, jum. यहाँ पे vector है flea. है ना? यहाँ पे vector है tick. यहाँ पे भी vector है Indian tick typhus. तो tick ही होगा ना? Tick दुम है. यहाँ पे mite. और यहाँ पे भी mite, not mite, the larva of the mite. Mite लिख के ब्रैकेट में लिखो चिगार पे बहुत क्वेश्चन आता है. The larva. So louse, flea, tick, mite. Total 6. Please learn the 6 together. Please learn. You can't afford missing that. सबके rash की peculiarity बताती हूँ. Come on the screen. So here the rash is everywhere except palm and sole. Please write down. है ना? Here the rash is trunk, followed by extremity. ठीक है, trunk followed by extremity. Here the rash is maculopapular, है ना, in RMS. ठीक है, here the Indian tick typhus, it is having ischar. ठीक है, rickettsial pox से मुझे याद आता है small pox, it is having vesicular rash. Yes, I'm coming on that point. Wait, vesicular rash, है ना, और Susugamushi में also ischar. ठीक है, rickettsial I forgot to tell you, this is most fatal, highly mortal, most fatal. So that you have to learn the peculiarity based on which you will get the clues in your question. Clues के लिए peculiarity याद करो. ठीक है, the last and the most important, the ultra important thing, the ultra super duper important thing is wheel phalanx. So please learn the results of the wheel phalanx, people. Please listen to my words. This one is OX19 positive, only OX19. This one is also सिर्फ OX19 positive. The next two, OX19 तो है ही, साथ में OX2 also positive, है ना, OX19 तो है ही, साथ में OX2 भी positive है ना. So this is Prowazaki, this is Typhi, this is Rickettsia rickettsii, this is Rickettsia conuri, है ना. Akari में everything is negative. Everything is negative. All of them are negative. Nothing, all three, nothing is positive. And Susugamushi में OXK is positive. Yes, this is how you have to learn. If you don't change the sequence, it's easy. है ना, पहले दो में सिर्फ 19 है, अगले दो में 19 के साथ 2 भी है, फिर सब कुछ नेगेटिव है और फिर OXK है. How many of you got it? Now I will show you the question. How many of you got it? Say yes or say no. Say something. Please make this table. You need time. Please take 1 minute and make it. I will give you 1 minute. Make it table. You get 100% questions of Rickettsia chapter is done. इसी में से आएगा और कुछ नहीं आएगा. I'm sorry. You can write drug of choice of all of these is doxy. कहीं भी लिख लो. Doxy. Doxycycline is DOC for all of them. Done. The chapter is done. Shall I remove it? Once you are done, kindly write down. Once it is removed, it will not come again. Shall I remove it? You have make it in your copy, or I will forward it to you. Shall I remove it? You can take the snapshot. Otherwise, लिखने का time नहीं है तो photo ले लो. Take some photo, take some snapshot, write down, but make it quick. ठीक है? RT rash, can you explain again? RT ka rash, it is trunk followed by extremity. First it is coming on the trunk and then it will come on the extremity. Okay. The disease of the Orientia Susugamushi is scrub typhus. Scrub typhus, the name of the disease, in which ischar compulsorily come. Okay, Lalit, anyone else want to ask anything? Please ask. Can I remove it? So someone have taken the snapshot and put it on the group also, the Telegram group. So please allow me to move now. Okay. Please turn round chair around them. Lo bhai. Where should I go? But okay, take the snapshot. Have you taken? Shall I come back now? Shall I come back now? Okay, ji. So shall I remove it? Give me a thumbs up. Quick, fast. Okay, done. So this is your table. Okay, now we will solve the question. Have a look on the question. You all will be able to solve that. Read the question, tell me the answer. Fatafat. Read the question, tell me the answer. There is a patient presented with rash all over the body, sparing palm and sole. Sparing palm and sole. Bas na ho gaya na question. Huh? The patient have no history of animal exposure. What can be the condition? Rashes everywhere except palm and sole. So of course, the answer is epidemic typhus. So you get many simple questions like that. Hai. The following is the agent for RMS, Rocky Mountain Spotted Fever. Rocky Mountain Spotted Fever किससे होता है? What is the agent? Is it Rickettsia rickettsii? Is it Rochimela quintana? Is it Rickettsia Susugamushi or Coxella burnetii? What's the correct answer? R for R. RMS is with Rickettsia rickettsii. You all are right. So you get many questions on that. Hana? Correct answer is A. You all are right. Very good. Okay, one more question. Read the question. There is a 25-year-old man went toward trekking, came back with rash. The wheel phalanx is OX2 and OX19. Two things are positive. OX19 is positive and OX2 is also positive. Now you tell me the diagnosis. OX19 and 2 is positive in which and which? You tell me. Anna, I told you 6 now. देखो अपना table. 6 में से 19 और 2 कहाँ पे positive? In the third and the fourth. Not first, not second. In the first and the second, only 19 is positive. In the third and the fourth. Who is the third and the fourth? The third is the Rickettsia rickettsii. The fourth is Rickettsia conuri. Among them, option have only Rickettsia rickettsii. If conuri was also there, it was also an option. It was also correct, I mean. But currently, my answer is Rickettsia rickettsii. It is the only option given here. Yes, absolutely right. Hannah, I'm changing this question. Currently, answer is A. You all are right. I'm changing this question. Listen, if I give only 19, not 2, then what is your answer? Only 19, not 2. What is your answer? The first two. The first two. In that case, Provozaki and typhi both will be the answer. Epidemic and endemic. Hannah, epidemic typhus, endemic typhus. Right? In that case, both will be your answer. RP, RT. A, B दोनों होगा ना? सिर्फ B क्यों बोल रहे हो? B and C दोनों होगा ना? It is present in B and C both, na people. Why you are saying only B? Yes, B and C both, है ना? So it is present in both. So यहाँ पे answer दोनों हो जाएगा. I'm changing the question again, people. I'm changing the question again. I'm giving you in the question not 2, not 19. It is OXK positive. What is your answer in that case? OXK तो सिर्फ एक ही में positive होता है. Only in one, scrub typhus. In that case, your answer will become scrub. So you get many questions on the wheel phalanx. Don't ever try to forget. If you don't change the sequence, everything is very good. And if you change the sequence, you will be in trouble. Read the question, tell me the answer. Wheel phalanx for scrub typhus is positive for what? OXK, OXK and 19, OX2 or OX19? I'm asking. What is the answer now? Yes, your answer is A. You all are right. Scrub typhus. I'm changing the question from scrub typhus to epidemic typhus. What is the answer now? You know my way. I keep on changing the question in multiple directions. So you think in every direction. What is the answer now? What is the answer now, people? Yes. If epidemic typhus, answer will become, see someone, answer will become D. Very good. If I change it to endemic typhus, still the answer is D. है ना? So this is how we approach. So wheel phalanx पे बहुत question आते हैं. ठीक है? Next question wheel phalanx पे based है. I'm not reading a big question based on the wheel phalanx result. You all can do that. You all can do that. है ना? You all can easily do that. देखो पूरी question में सिर्फ एक word है tick. काम का एक ही word है tick. Tick वाले का wheel phalanx क्या है? They want to ask. है ना? Read the question by yourself. बकवास बकवास बकवास. The only word which is important there is tick. I have marked that. है ना? Tick वाले का wheel phalanx क्या है? Tick कहाँ पे आता है? So you tell me the vectors. 6 के vectors क्या हैं? Louse, flea, tick, mite or larva of mite. This is the sequence now. So tick come in 2. Which two? The third and the fourth. So third and fourth का wheel phalanx क्या है? Third and the fourth का. They want to ask that. So third and fourth का wheel phalanx है 19 and 2 both positive. So correct answer is D. Yes, you all are right. This is the approach. This is the approach. You got it? Does anyone have any confusion? Can I proceed ahead? Can I proceed ahead, people? Huh? Okay, few more questions. Drug of choice. In सबकी drug of choice is doxycycline. Okay. Okay, the next question. Drug of choice is doxycycline. Okay, ji. The next question. Scrub typhus caused by Orientia Susugamushi is transmitted by. They want to ask the vector. The vector of the scrub typhus. Is it flea, chigar, tick, or mosquito? What's the scrub typhus? What is the correct answer, people? Fast. It is chigar. Chigar, it's repeated PYQ. The larva of the mite. The larva of the mite. है ना? If I change the question to endemic typhus. What is the answer now? Endemic typhus में answer क्या हो जाएगा? बोलो. Endemic typhus is the second one. दूसरा वाला. You learn the sequence. It's very easy if you learn the sequence. Yes, in that case answer will become flea. है ना? एंडेमिक टाइफस कर दूंगी तो फ्लिया हो जाएगा. और यहाँ पे अगर मैं रॉकी माउंटेड स्पॉटेड या इंडियन टिक टाइफस कर दूं तो आंसर विल बी टिक. So in this way you get many questions on the vectors also. Okay, so we are done with the rickettsia. Let me go ahead. So this rickettsia is also done. Let me come back on my PPT. Okay, so we are done with rickettsia. Just a second. So let me come on the next organism. Let me come on the next bacteria. So does anyone have any doubt here in the rickettsia? Can I move to the next organism? The next organism, I'm teaching you the miscellaneous one, people. The 4 miscellaneous were pending now. You asked me you want to study the miscellaneous also. So I am teaching you the miscellaneous one. Miscellaneous में we have already completed rickettsia. ठीक है, and now I am coming on mycoplasma. Now I am coming on mycoplasma. So let's start mycoplasma. The next one is the mycoplasma. The next one is the mycoplasma. We are done with the rickettsia and now I'm coming on mycoplasma. So mycoplasma. ठीक है? Mycoplasma is the only bacteria which don't have cell wall. I told you yesterday, the only natural bacteria which don't have cell wall is the mycoplasma. That's why they are known as jokers. They can change their shape very well. In the mycoplasma family, there are two bacteria, mycoplasma and ureaplasma, both are there. They don't have cell wall. They are non-capsulated, non-sporing, non-motile. Still, they can move with gliding motility. We don't know the mechanism, how does they move. Yesterday, I taught you 12 bacteria. 6 are motile by polar flagella, 6 by peritracheus. Mycoplasma is neither coming here nor coming here. So neither it is having peritracheus flagella, nor it is having polar flagella. It doesn't have any flagella. So ideally, it should be non-motile, but still it moves with gliding motility. We don't know the mechanism. We don't know the mechanism. Okay, we don't know the mechanism. It don't have a cell wall. It don't have a rigid cell wall. It don't have any cell wall. Have you heard about the beta-lactam antibiotics? Have you heard about beta-lactam antibiotics? What are beta-lactam antibiotics? The beta-lactam antibiotics are penicillin, cephalosporin, monobactam, carbapenem. How does they act? How does these all act? These all antibiotics act by inhibiting the cell wall. लेकिन इसके पास तो cell wall ही नहीं है. So I mean to say this bacteria is not killed by beta-lactam antibiotics. Because what is the mechanism? Beta-lactam antibiotics, that is penicillin, cephalosporin, they kill the bacteria by inhibiting the cell wall. But mycoplasma don't have the cell wall. Mycoplasma don't have the cell wall. So it is not killed by beta-lactam antibiotics. Say yes. Say yes. Okay. What was Gram stain? What was Gram stain? Gram stain, in the Gram stain, we said the principle of the Gram stain is the peptidoglycan content of the cell wall. If the peptidoglycan content is more, it is Gram positive. If the peptidoglycan content is less, it's Gram negative. Have I taught you yesterday? लेकिन इसके पास तो cell wall ही नहीं है. No peptidoglycan. क्या ज़्यादा क्या कम? No peptidoglycan. We don't use Gram staining here. That's why we use Giemsa staining. So it doesn't have cell wall. It doesn't have cell wall. No cell wall. Since it doesn't have cell wall, they can change their shape to cocci also, bacilli also. They are pleomorphic. So the point is that I'm teaching you a new chapter, mycoplasma, a bacteria, mycoplasma. It's a bacteria. The point is that it don't have the cell wall. Since it don't have the cell wall, there are three consequences. Number one, it can change its shape very frequently. Since it can change its shape very frequently, it is known as pleomorphic. Pleomorphic. Since it don't have the cell wall, we cannot use Gram stain in that. Because there is no cell wall. The basis of the Gram stain is cell wall. But no cell wall, we can't Gram stain. We use Giemsa stain. We use Giemsa stain. Number three, since it don't have the cell wall, it is not killed by beta-lactam. Beta-lactam are resistant. Because the mechanism of beta-lactam is inhibiting cell wall. But since they don't have the cell wall, they are resistant to beta-lactam. So please learn the three things. Say people yes. Say yes. This is mycoplasma. Mycoplasma, they don't have the cell wall. Please learn its culture. Please learn its culture. The Selactomedia is PPLO. What is the Selactomedia? Say it out. PPLO is the Selactomedia, on which the appearance of the colony is fried egg. Can you see fried egg? Have you ever tried hatching an egg on a pan? So we see a yellow at the center and white at the periphery. देखा है egg? Egg कैसा दिखता है? हाँ? Egg है. So this is the egg. It is looking like a fried egg appearance. It is looking like fried egg appearance. Please learn. It is an image-based question also. Mycoplasma. The Selactomedia is PPLO. It's a fried egg appearance. It causes basically pneumonia. It causes basically pneumonia. URTI and pneumonia. Pneumonia. The pneumonia which is caused by mycoplasma is known as Eaton agent. Eaton agent pneumonia, atypical pneumonia, walking pneumonia. So it's pneumonia. It's pneumonia. We are done. Very small chapter. Read the question, tell me the answer. In reference to mycoplasma, all are true except. So what is false? What is false for mycoplasma? Can you tell me what is false? Which of the following statement is false? Which of the following statement is false? They are inhibited by penicillin. They reproduce in cell-free media. They have affinity for the membrane and they can pass through 450 nanometer. Yes, A is false. They can never be inhibited by penicillin. No, they are resistant to penicillin because they don't have cell wall. So this question is repeated PYQ in various languages. The concept is same. We cannot kill mycoplasma. We cannot kill mycoplasma by using beta-lactam antibiotics. Any beta-lactam. We cannot. Please learn the concept. So we are done. Coming on the next chapter, chlamydia. The last chapter we are coming is chlamydia. Chlamydia and spirochetes. So let's come on the chlamydia. The next chapter is the chlamydia. Family Chlamydiaceae. Let me start the family Chlamydiaceae. Here we have three chlamydia: chlamydia trachomatis, psittacosis, and pneumonia. But psittacosis and pneumonia we don't have to read. Psittacosis causes disease in birds, parrot, bird में कराती है. We don't have to study. And pneumonia causes only pneumonia. So basically, I have to teach you chlamydia trachomatis. What I have to teach you? Chlamydia trachomatis. There are 18 type of chlamydia trachomatis. कितने तरह का होता है? 18, 18, 18 type. है ना? Again, you have to learn one table. That's it. है ना? 18 type of chlamydia. 18 type of chlamydia trachomatis. How are the 18 type? 18 type, the first four, A, B, B, A, and C. The first four causes trachoma. It causes only one disease, trachoma. The last three, L1, L2, L3, they causes only one disease, LGV, lymphogranuloma vanorum. It is an STD. It is an STD, sexually transmitted disease. The remaining all causes three disease. They cause three disease: genital chlamydiasis, inclusion conjunctivitis, and infant pneumonia. Got it? So please learn total 18 types. Please learn total 18 types. 18 types of chlamydia. What are the 18 types of chlamydia trachomatis? Say, ma'am, A, B, BA, and C. Four are these. No problem. L1, L2, L3. Three are these. No problem. So tell me one disease and the remaining D2K. remaining D2K. Learn like this. Total, if you count, they are 18 in number. Hai? So say the diseases caused by them. Who will tell the diseases? They cause only one disease, trachoma. Khatam. One disease, bol ke khatam karo. Trachoma. They cause one disease, bol ke khatam karo. LGV. Bol ke khatam karo. LGV. And they cause three diseases. You should be able to tell me the name of the three diseases. What are the three diseases? एक तो आई की है. देखो ये आई की है ना, तो यहाँ पे भी एक आई की है. इंक्लूजन कंजंक्टिवाइटिस है ना, ये जेनाइटल है ना, तो एक यहाँ पे भी जेनाइटल है, जेनाइटल क्लैमाइडियासिस है ना. सो वन इज ऑफ आई, वन इज ऑफ जेनाइटल, एंड वन इज निमोनिया. वन इज निमोनिया, infant pneumonia. So one eye, one genital, and one pneumonia. Okay, so please learn. So these are the diseases. Maximum question are from here only. So which serotype causes what? A1, A, B, BA, and C causes trachoma. L1, L2, L3 causes LGV. D2K causes three diseases. You should know the name. Okay, so let's start with A, B, B, A, and C. They causes trachoma. Have you read trachoma and ophthal? Have you read trachoma and ophthal? Say yes or say no. You may have read it, no? It is a cause of blindness. It can cause blindness in India. There are follicular hypertrophy. You can see the follicular hypertrophy is there. Pannus is there. Safe strategy पढ़ी है PSM में. Have you read PSM? PSM में what is safe strategy? You know. So S is surgery, antibiotic, facial hygiene, and environmental hygiene. So that is the safe strategy. I guess you have read it. So trachoma is not a topic in microbiology, and it is a topic in ophthal. It is a topic in PSM. But here I want to tell कि trachoma karata कौन है? A, B, B, A, C. That you have to learn from micro point of view. है ना? And there are inclusion body. इसके अंदर if you take the biopsy, you will get the inclusion body. है ना? So inclusion body का नाम है HP body. You can see the inclusion body. है ना? In the trachoma. HP body. HP body is Hellbell-Storer Provozak body. है ना? Hellbert-Stodler Provozak body. You can see HP body also. है ना? Because you don't have to write the spelling. You have to identify in the options. It is HP body. है ना? So we are done with A. A, B, B, A, C. The next is D2K. The next is D2K. So can I start D2K? It causes three diseases. Genital chlamydiasis. In the males, it causes urethritis. In the females, it causes cervicitis. Learn the most common. जैसे कल gonorrhea में था, same like gonorrhea. Males, it causes urethritis. Urethritis, it causes proctitis, it causes epididymitis. In female, it causes cervicitis. Endometritis, salpingitis. Learn the first. Here it's urethritis, here it's cervicitis, like gonorrhea only. Like gonorrhea only. So can we go ahead? So this is genital. Inclusion conjunctivitis, it is the conjunctivitis you will read in ophthal, and pneumonia. And L1, L2, L3 causes an STD. The name of the STD is lymphogranuloma vanorum. It causes lymphogranuloma vanorum. Okay, it is an STD. We do a Fry's test. It is a skin test to diagnose it. L1, L2, L3 mein maximum it is caused by L2. It's also a question. It's not L1. Maximum is L2. Maximum is L2. Can you see? This is male, the penis of a male, the vulva of a female. Again, male and female. You can see in LGV the genital ulcer. Can you see this is genital ulcer? Say yes. Can you see these are the genital ulcer? Say yes. And these are the enlarged lymph nodes. Along with ulcer, the enlarged lymph nodes. The enlarged lymph nodes are known as buboes. Like the plague. These are known as buboes. So enlarged lymph nodes are there. In male also, the female also, the inguinal lymph nodes are enlarged, which are known as bubo. The point is that the ulcer is painless, the lymph nodes are painful. Very unique. I'm saying the ulcer is painless. There is no pain in ulcer. And the lymph nodes are painful. The lymph nodes are painful. The deadly combination. Painless genital ulcer with painful bubo. Say it out. Say it once. Painless genital ulcer with painful bubo. बोलो अल्सर में दर्द नहीं है, पेन नहीं है, लेकिन ब्यूबो में दर्द है. So this combination is LGV. It's typical LGV. It's the biggest clue given to you. है ना? So that is the thing. Okay. So read the question and tell me the answer. Read the question. Lymphogranuloma vanorum is caused by which organism? It is Chlamydia trachomatis. The serovar L1, L2, L3. So there are not much questions from this. Price test is for LGV. We are done. The last is the spirochetes. Spirochetes are the thinnest bacteria. है ना? Bubo, can we say inguinal hernia? No, Abhijit. Bubo is a lymph node. It's not hernia. It's never hernia. Bubo is a lymph node, inguinal lymph node, which is enlarged. Coming on the last bacteria, finally last bacteria, it's spirochete. Spirochetes are the motile bacteria. What are spirochetes? They are the motile bacteria. They are very thin. They are very thin. They are like the thread. So there are three type of spirochetes: Trypanosoma, Borrelia, and Leptospira. Okay? So you have to learn the species and the diseases caused by them. That's it. So Trypanosoma, Borrelia, and Leptospira. In the trypanoma, there are four types of trypanoma. Trypanoma pallidum, which causes syphilis, and it is a sexually transmitted disease. Trypanoma pertue, it causes yaws. Trypanoma endemicium, it causes endemic syphilis, endemicum. And Trypanoma keratium, it causes pinta. So learn the trypanoma and the corresponding disease. You get a direct question on that. There are also four Borrelia. Borrelia recurrentis causing relapsing fever. Borrelia dutonti causes relapsing fever. Borrelia Brugdolferi causes Lyme's disease, very common PYQ. Borrelia vincenti causes Vincent angina. ठीक है, लेप्टोस्पायरा में एक ही है, एंट्रोगन्स. It causes leptospirosis, which is via rodent urine. It's also a PYQ. So your complete table में से ही सारे क्वेश्चन आएंगे, all the PYQs. So you just learn that, है ना? So we are done with that also. ठीक है? So finally we are done with bacteriology. How many of you are satisfied with the bacteriology? We are done with complete bacteria. I have not skipped even a single bacteria. All your PYQs will be from my notes only, that I take the guarantee. So just read my notes, blindly trust it. Your entire question of the bacteriology will be from this. I have provided you the charts also. You want elaborated notes that are provided to you. You want the charts that are provided to you. We are done with bacteriology. Anna, I wish ये कल ही complete हो गया होता. But yesterday I have to leave early. That's why I couldn't complete it. But now it's completed. So all the bacteria are completed. All till now. Nothing is left. Everyone is with me. Anyone having any doubt? You want something to repeat or you have any doubt? I mean, okay. So everything is completed in bacteria. So till now, complete general is done. Complete bacteria is done, and immuno may except few topics, immuno is also done. Whatever is left in the immuno, we will take it to today evening or tomorrow, but I will complete. Don't worry. Okay, charts are little bit incomplete. The small bacteria are not covered, Usman. The big bacteria are there. So for last time revision, they are sufficient. Okay, the small bacteria are not covered. The small में चार तो लाइनें हैं that you have to learn. Okay. Among these, they all have equal importance. I cannot say, but yeah, they have the equal importance, especially gram-positive cocci and gram-negative bacilli. You get many questions, but you have to read all. Anyways, you cannot skip. Yes, if you forgot, you can read the charts only. And timely charts are the best. Have you read it? Have you tried it after the lecture? Have you tried it? So please read the charts. Hannah, it is the exact replica whatever I taught you. So can I start virology now? Can we start the virology? Say yes, people. So we are done with this. Let me open another PPT. The another PPT is the virology, a new beginning, the virology. Okay. So let me start with virology with full energy, with full dedication. Everyone with me. Everyone is with me. So can we start viro? Can we start viro? No, I'm not giving the break. I will give you a direct lunch break, which will be little bit earlier. Not lunch or whatever you say. Maybe after half an hour. At 12:15, I will give you the break. From 12:15 onwards, I will give you a large break, maybe 40 minutes. ठीक है? So let's continue. Till now, general virology हो जाएगा. ठीक है? अभी है half an hour or 1 hour, whatever. ठीक है? चलो, let's start. Let's start. Okay? So let's start with virology. Parasite I will take tomorrow. I want all of you to be interactive. Please, every time you will keep on answering. let's start virology. ठीक है, ठीक है. So let's start virology. Before starting virology, you must know the structure of a virus. How does a virus look like? What is the page number? What is the page number, please? Can anyone tell the page number? Please, everyone open. Everything is written in your notes. You don't have to write anything. Entire virology is given in the notes, I guess. What's the page number? Can anyone share the page number, please? Anyone? 23. So please open page number 23 onwards. Everything is given. So just now open it and please, whatever I'm asking, answer that only. Don't talk. Talk with me only. Listen, chalo, let's start the structure of a virus. How does a virus look like? All the virus in this world, there are two types of virus. But all the virus, one thing in common, they have the nucleic acid. All the virus have nucleic acid. Mind my words. What is the nucleic acid? It can be DNA or RNA, never both. Human cell contain both. My cell have DNA also, RNA also. But virus have either DNA or RNA, never both. Never both. So viruses have either DNA or RNA, never both. Mind my words. All viruses have nucleic acid. So this is the nucleic acid, number one. Number two, all the viruses have a protein covering around the nucleic acid. Protein covering. This protein का जो covering है ना, this is known as capsid. It is known as capsid. Capsid. The capsid is the protein covering around the nucleic acid. This is a polymer. It is made up of monomer. The monomers are capsomer. These all are monomer. The monomers are known as capsomer. Capsomer make capsid. So capsid is made up of capsomer. How many of you got it? Capsid is made up of capsomer. Capsomer are the monomer. They bind together to make a capsid. Capsid is a protein covering. It is also present in all viruses. So these are the two things present in all viruses of the world. Now there are third thing which is not compulsory. It is present in some virus, absent in others. That is envelope. Some viruses have envelope, some do not have it. Got my point? The viruses which have the envelope, they are known as enveloped virus. The viruses which do not have envelope, they are known as naked virus. They are known as naked. Don't say non-enveloped, say naked. So there are two type of viruses, naked and envelope. And envelope is not compulsory, I'm saying. But the nucleic acid present in both, capsid present in both. Some have envelope, some do not have it. The one which have are enveloped. The one which do not have are naked. They are known as naked. That is the third thing. And the fourth thing, on the envelope, sometimes there are spiky projections. Spiky projections, again not compulsory. It is known as papilloma. So see, have a look on the diagram. Can you see the two viruses? Virus 1, virus 2. See the two viruses in front of you. Both the viruses have one thing in common, nucleic acid. The nucleic acid. Both the viruses have one thing more common, capsid. Can you appreciate the capsid in both of them? This is capsid made up of capsomer. This is capsid made up of capsomer. But the first virus is naked, don't have the envelope. But the second virus, apart from the capsid, it is having a protein covering also. That is known as envelope. The envelope is having the papillomas. The spiky projections, that is the envelope. Got my point? Say yes. So capsid is made up of capsomer. The capsid is made up of capsomer. Okay? Can we go ahead? This is the structure of the virus. So you didn't ask me, ma'am, nucleic acid is made up of nucleic acid, that is DNA or RNA. This is the composition. DNA or RNA, we got it. Capsid is made up of protein. Capsomer, that is protein, we got it. But what is the composition of envelope? Envelope किस चीज का बना है? Envelope किस चीज का बना है? ये DNA RNA का बना है, ये protein का बना है. What is the composition of envelope, people? Envelope is made up of two things, lipoprotein. Not only protein, lipoprotein, lipid and protein. So envelope is made up of two things, lipoprotein. Composition of envelope, this envelope. Envelope is made up of lipid protein, lipoprotein. Lipid host का चुराया है, host is human, हमारा चुरा के, और protein अपना खुद का है. So protein is of virus own, but the lipid is the stolen, चुराया हुआ है. किसका? Host. It is host derived. So that is lipoprotein. Got my point? Read the question, tell me the answer, quick, fast. What is capsid in the virus? What is capsid in the virus? Is it extracellular infectious particle? Is it protein covering around the nucleic acid? Is it envelope, or is it none of the above? What is capsid? What is capsid? Yes, it is the protein covering. It is a protein covering. So let me classify virus. Before starting the virology, you may be knowing there are two types of virus. DNA virus, RNA virus. I told you all the viruses have either DNA or RNA, never both. Never both. Never both. So how many viruses are DNA? How many viruses are RNA? So this is the basis of classification. What is the basis of classification in bacteriology? You will see now gram staining. We have 4 type of bacteria. We have seen na GPC, GNC, GPB, GNB. So that was the basis of classification there in bacteriology. Here in virology, we have 2 type of virus, DNA virus and RNA virus. Say yes, the DNA viruses and RNA viruses. There are 6 family of DNA virus having multiple members inside that. There are 15 family of RNA viruses having multiple members inside that. Got my point? So these are the family having multiple members. I will give you 2 mnemonic now. DNA, RNA. So DNA viruses, the 6th family. The 6th family of DNA virus, the mnemonic is double H A triple P. Double H A triple P. And you can say happy. Double H A triple P. What is double H? One H is herpes, one H is hepadina. Bolo, herpes and hepadina. Double H. A ki hai, only one A, adeno. Only one A, adeno. And it's triple P. It's triple P. है ना? Pox, parvo, papova. Pox, parvo, papova. है ना? Please understand, in virology, P is very dangerous. I'm also P. है ना? So don't underestimate any P. ठीक है? P बहुत dangerous है. Why it is dangerous? Listen, there are 3 DNA viruses which start from P. There are 2 RNA viruses which start from P. So 3 DNA and 2 RNA, total 5 bacteria, 5 viruses which are P. And you will always get a confusion between them. I know. So please learn the 3 P here very clearly. Pox, parvo and papova. Pox, parvo and papova. Don't forget. Okay? You can learn the members also inside each family. Don't learn the family, learn the members inside the family. I know. But I'm not reading it right now, the members, because one by one I will teach you all 6 now, today only. I know. So whenever I start a family, then I will tell the members and then I will explain. Whenever I will start a family, I will tell the members and then I will explain. So I'm not reading the members here. Anna, I will teach you all 6. Don't worry. Today only. So first I will start with pox only. Today only. Okay? Abhi hum classification ke baad we will start. Okay, got my point? So six families of the DNA viruses. Anna, there are 15 families of RNA. Let me come on RNA. There are 15 families of RNA. I'm having a mnemonic for that. What is the mnemonic? The mnemonic is ABCD. FOR for TPH, tweet per hour, or make it PTH, parath hormone. है ना? ABCD for TPH, make a mnemonic. है ना? So there is A is arena. वहां का A क्या था? Adeno. और यहाँ का A क्या है? Arena. B is bunia. है ना? There are 2 C, corona and Kelsey and corona. You can see the 2 C. है ना? There is only one D. D is delta. There are 2 F, philo and flavi. है ना? See, where is philo, where is flavi and philo. There are 2 F. O is orthomyxo. O से एक ही होता है. Orthomyxo, है ना? O is orthomyxo. There is triple R. You have to learn triple R. Ultra important. तीनों R. है ना? Reovirus, retrovirus and rhabdovirus. मैं तीनों पढ़ाऊंगी. I will teach you all R today only. तीनों R. है ना? T is toga. Only one T is there, toga virus. है ना? There are 2 P. Don't P, है ना? P को light P, because there you get many question on the P. P में बहुत confusion होगा. I will show you the question also. Wait. What are the 2 P? Paramyxo and picorna. And last is H. H is hepe. ठीक है? Listen. I will make it easy. Don't worry. At the first look, it may look difficult to you, but it's not difficult. It's easy. ठीक है? DNA virus, RNA virus. ठीक है? Tell me how many families we have. You will see, ma'am, 6 families of the DNA and 15 family of the RNA. Family. First, say the name of the family. Here the mnemonic is double H A triple P, and here the mnemonic is A B C D. Make 2 C. FOR, 2F and 3R, and TPH. Make 2. Count, they are 15. You can count if you wish. ये 15 है ना? Yes, you have to learn this mnemonic, है ना? You have to learn the full form. If you can't learn, आगे कुछ नहीं समझने आने वाला. The basic thing, मैं किसी वायरस का नाम बोलूं तो you should have orientation कि man is talking about a DNA virus or RNA virus. If you don't know even that, so reading virology is not good for you, है ना? So first thing, you should understand the classification. We will study the virus by virus, but you should have orientation which is DNA virus, which is RNA virus. If you don't know the gram staining in the bacteria, yesterday I taught you the basic classification. Now, if you don't know that classification, how you can understand the entire bacteriology? So this is basic, है ना? So what is double H? Say the full form once. बोलना है एक बार. It's herpes. It's hepadina. Herpes and hepadina. A is adeno. Here A is arena. Arena. है ना? The triple P here. It's pox, parvo, papova. Pox, parvo, papova. Here also we have 2 P. It's picorna and paramyxo. बोल के याद होंगे, बोलना पड़ेगा, है ना? B is Bunia. The 2C is Kelsey and Corona. D is Delta. The 2F is Philo and Flavi. O is Orthomyxo. Triple R is very important. Rio, Retro, Rhabdo. T is Toga virus and H is Hepe. If you know the mnemonic, saying it is very easy. Difficult नहीं है, बहुत easy है. समझ में आया क्या? Okay, someone is giving some mnemonics for that, है ना? You can understand the mnemonic. I'm coming on these mnemonic naked और सब पे आ रही हूँ मैं. Arshad, I'm coming on all these points. But first thing is to learn the classification. ठीक है? You learn the classification. Now you should know the members inside each family. Inside each family, members भी देखने, but I'm not reading it. One by one, I will take it and teach you. So from here, I will teach you 6. So here, I will not teach you all 15. The 6 important we will say. 6 from here, 6 from here. 12 viruses I have to teach you today. Are you people ready? Virology is a small section. We will easily complete it till evening. I know. So virology is very small section. Say yes. Okay. So total 12 viruses, 6 from the DNA, 6 from the RNA we are going to complete. If you know the classification, everyone know the classification? Can you answer this question? Very simple question. Which of the following is not a DNA virus? Say the mnemonic. DNA का mnemonic क्या है? It's double H A triple P. What is not coming in this mnemonic? Parvo coming or not coming? You will say, ma'am, one of the P is parvo. Papova, one of the P is papova. Pox, one of the P is pox. But rhabdovirus is not coming here. The rhabdovirus is coming in RNA viruses. A, B, C, D, F, O, R का one of the R है वो. So the correct answer is rhabdo. So you get very simple questions based on the classification. You cannot afford missing that. ठीक है, listen. Based on the classification, I want to tell you 4 important points. You can't afford missing that. एक एक करके बताऊंगी. चारों point लिख लो, रट लो. Please learn that. Listen, DNA virus, RNA virus. I have to write this mnemonic 4 times. I will not say the full form now. Full form, I'm assuming you know that. I will not say the full form, but I will write the mnemonic. Double H A triple P, है ना? And the RNA virus is A B C D F O R T P H. ठीक है? Now listen what I want to tell you. Listen everyone. You tell me DNA is double stranded or single stranded? RNA is double stranded or single stranded? Everyone knows that normally. Normally DNA double stranded होता है कि single stranded होता है? DNA is always double stranded. And RNA is always single stranded. Everyone knows? I guess everyone knows this basic. है ना? ये तो सबको पता है कि नहीं? So all DNA viruses are double stranded because they contain DNA. Can I say all the 6 DNA viruses are double stranded? Yes. They all are double stranded except one. You have to learn the exception. Except one. One of the P, that is parvovirus, which is DNA but single stranded DNA. Which is DNA but single stranded DNA is very unique. Normally we don't have single stranded DNA, we have double stranded DNA. So they all have double stranded DNA except parvo. Learn the exception parvo. I want you to learn the exception parvo. Please learn. Please learn the exception parvo. Okay. So can I come on the RNA? All the RNA viruses are single-stranded. Since RNA is a single-stranded molecule, so all the RNA are single-stranded except one exception, one of the R. That is Rio. Rio is RNA, but it's double-stranded RNA. It's double-stranded. RNA is never double-stranded, but in Rio it is double-stranded. Learn the 2 exception. I want all of you to learn the 2 exception. What are the 2 exceptions, please? Parvovirus and Riovirus. Please learn the 2 exceptions. Got it? So the summary is that all DNA are double-stranded except parvo, which is single-stranded DNA. All RNA are single-stranded except Rio, which is double-stranded RNA. Got this point? The same thing is written in your notes. Mark it. All DNA are double-stranded except parvo, which is single-stranded DNA. All RNA are single-stranded RNA except Rio, which is double-stranded RNA. Say yes, people. Say yes. Read the question. Tell me the answer. फटाफट दोनों PYQ हैं. Both of them are PYQ. Read the question. देखो, it is a PYQ of FMG only. Which of the following viruses single-stranded DNA? I am asking a unique thing. DNA is never single-stranded. Only in one it is single-stranded. What is the correct answer? Yes, the correct answer is parvo. It's a PYQ. You can't afford missing that. ठीक है? Of course, everyone knows the answer is parvo. ठीक है? Please everyone answer. ठीक है? The next question. Which of the following is a double-stranded RNA? See, again a unique thing. RNA is never double-stranded, but in one of the virus it is double-stranded. I want to know the name of that virus. Is it Rio, Retro, Rhabdo, or Picorna? See, the question is very confusing. So what is the answer? Here also answer is Rio. The answer is A. And also you have to learn that. You have to learn that. ओके चलो अब अगली चीज you tell me we have DNA viruses, RNA viruses. Let me write it again. DNA and RNA. In short, I will write. I have to write it 4 times, है ना? Because I want to tell you 4 things in that. So double H, A, triple P, and here we have A, B, C, D, F, O, R, T, P, H. ठीक है? Okay, learn the full form. Now let me ask you 6 questions. Tell me smallest DNA. Among the DNA, who is the smallest? And smallest RNA. Among the RNA, who is smallest? And smallest overall. है ना? इन दोनों में से कौन सबसे smallest? DNA और RNA overall smallest कौन है? ठीक है? Tell me the three. These three. Then tell me who is the largest DNA. है ना? Tell me who is the largest RNA. ठीक है? The mnemonics are in front of you. Largest RNA. उन्हीं में से I will pick and tell me the largest overall. Largest overall. Wait. I will come. You got my 6th question. First, have a look on my 6th question. First, you all have a look on my 6th question. है ना? Smallest DNA, smallest RNA, smallest overall. Largest DNA, largest RNA, largest overall. You got my point. है ना? Let me give you a clue. Very good clue. They all are P. I told you, na, don't underestimate. P. No real confusion. A real confusion between the P. ठीक है? This is a clue or a confusion, whatever you say. ठीक है? Let me help you in explaining. चलो वापस आओ सब. Come back, everyone. Among the DNA, who is the baby of this family? This is a family, no? Who is the newborn? छोटा सा baby कौन है इस पूरी family का? Smallest कौन है? Answer is Parvo यहाँ का baby है. Parvo से I imagine a little girl, cute girl, Parvo. Little baby, newborn baby. छोटी सी baby है, Parvo. है ना? Boy, girl, whatever. ठीक है? It's a newborn. Parvo से मुझे छोटा सा imagine होता है. ठीक है? Who is the grandfather of this family? Grandfather, the senior member of this family. Complete DNA में से. Answer is Pox. I'm sorry, I should write Pox here. है ना? The largest DNA. Pox is the grandfather. So इस family में the baby is Parvo and the grandfather is Pox. नाम से कैसा लग रहा है? Pox. He is the grandfather. और Parvo is a cute baby. Just I'm helping you to learn. Otherwise PP सब confuse हो जाएगा. पार्वो को इमेजिन करो एक छोटी सी बेबी है और पॉक्स को इमेजिन करो एक सीनियर एकदम एजेड 90 ईयर 100 ईयर के ग्रैंडफादर है. ही इज द पॉक्स. नाम है उसका पॉक्स. ठीक है? चलो कम ऑन द आरएनए. आरएनए में हु इज द बेबी? इस फैमिली का बेबी कौन है? हु इज द बेबी ऑफ आरएनए? आरएनए में बेबी है पिकोरना. पिकोग्राम क्या होता है? नैनो, पिको, डेको. पिको मतलब होता है 10 पर -12. यू हर्ड पिकोग्राम, नैनोग्राम. हाँ ऐसा सुना है? तो पिको वहीं से आया है. पिको मतलब इतना छोटा कि 10 to the power -12. तो वहां से याद आएगा तुम्हें पिकोरना इज पिको. ये यहाँ की बेबी है. वहां की बेबी है पार्वो. यहाँ की बेबी है पिको. देखो दो बेबीज हैं, पार्वो, पिको. पार्वो इज द बेबी ऑफ डीएनए एंड पिको इज द बेबी ऑफ आरएनए. वहां का ग्रैंडफादर था पॉक्स. हु इज द ग्रैंडफादर हियर? यहाँ का ग्रैंडफादर है पैरामिक्सो से मुझे कैसा याद आता है? मिक्सो के बड़े भैया मिक्सो के बड़े भाई है ना? बड़े भाई है ना? ही इज द सीनियर. सो राइट डाउन पैरामिक्सो. यहाँ के ग्रैंडफादर है पैरामिक्सो, बड़े भैया है पैरामिक्सो. गॉट माय पॉइंट? अब देखो एवरीवन ऑन द स्क्रीन. चलो यहाँ की बेबी है पार्वो, यहाँ की है पिकोरना. तो दोनों स्मॉल है, लेकिन ओवरऑल स्मॉल कौन है? सबसे छोटी कौन है? पार्वो छोटी है कि पिकोरना छोटी है? तो आंसर इज पार्वो. जो डीएनए है ना दैट इज ओवरऑल भी पार्वो ही छोटी है. दोनों ग्रैंडफादर को कंपेयर करो. वहां का ग्रैंडफादर है पॉक्स, यहाँ का ग्रैंडफादर है पैरामिक्सो. सो हु इज मोर सीनियर? बोथ आर सीनियर इन देयर ओन फैमिली, बट हु इज मोर सीनियर? सो आंसर इज पॉक्स ओनली. सो जो डीएनए में है द डीएनए वन आर ओवरऑल ऑल्सो. गॉट माय पॉइंट? शैल आई रिमूव दिस पेज and the same thing is written in front of you. You will tell the answers. Chalo, you will tell me. You will tell the answers now. Smallest DNA, smallest RNA, smallest overall. है ना? The 3 questions in front of you. And again you tell me largest DNA, largest RNA and largest overall. ठीक है? Again the 6 questions in front of you. Let me give you a clue. P. Start writing the question in this sequence. So first, DNA का बेबी कौन है? पार्वो. है ना? RNA का बेबी कौन है? पिकोरना. है ना? दोनों में से छोटा बेबी कौन है? ज्यादा छोटा कौन है? ये जस्ट न्यूबॉर्न है पार्वो. सो पार्वो है ना? मे बी ये इन्फेंट है, वो न्यूबॉर्न है. सो इट इज पार्वो ओनली. ग्रैंडफादर्स बताओ. डीएनए का ग्रैंडफादर कौन है? पॉक्स. आरएनए का ग्रैंडफादर कौन है? पैरामिक्सो और ओवरऑल ग्रैंडफादर कौन है? तो पॉक्स और पैरामिक्सो दोनों बड़े हैं, मे बी ये 100 ईयर्स का, ये 80 ईयर्स का, तो ज्यादा सीनियर कौन है? पॉक्स. लर्न लाइक दैट, प्लीज लर्न. These are your 6 PYQs we have covered. Say yes, people. So same thing is written in front of you in your notes. Say yes, say something, react. Now you will not forget. So everything is P, 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 P. Still you don't forget. Still you can learn. Read the question, tell me the answer. The virus with the smallest genome. I have not written DNA or RNA. I am asking overall. I am asking overall. If I have not written DNA or RNA, I am asking overall. Tell me the smallest virus overall. Overall smallest. सबसे छोटा baby. Who's the baby? The baby virus. The baby virus. It's parvo. Very good. The correct answer is parvo. I am changing the question. I am including the word DNA. Would you like to change your answer or still same? I'm including a word DNA. Would you like to change the answer, yes or no, or still same? Answer is still same. It's still same. Answer is still same. है ना DNA में. If I change it to RNA, then what is your answer now? Now to change karo. Now you should change it. Smallest RNA. What is the answer in that case? Yes. In that case, answer will become picorna RNA में. So you get questions on that. Chalo. Chalo, chalo. Everyone on the screen. I told you DNA viruses and RNA viruses. Let me divide. DNA and RNA, है ना? चलो मैं तीसरी बार बता रही हूँ. एक चीज मैंने बताई सिंगल स्ट्रैंडेड डबल स्ट्रैंडेड. One thing I told you single stranded double stranded वाला concept. The second thing I told you smallest largest वाला concept. The third I am telling you envelope non envelope का concept. And fourth I will tell you segmented. 4 concepts I have to tell you in the classification. है ना? So first concept I guess everyone know. Everyone know the first concept. Just a second. है ना? That single stranded we will revise in the end. चलो DNA viruses RNA viruses write down. I will tell you the envelope. DNA RNA we have to write it once more. I have to write it 4 times, है ना? So double H A triple P, and here we have ABCD or TPH like this, है ना? चलो. Here we have total 6. Here we have total 15. I told you all viruses are not enveloped. Some are enveloped, some are naked. So how many are enveloped here? How many are naked? Don't use the word non-enveloped. I like the word naked because I will correlate something. Say naked. Non-enveloped is naked only. You can say non-enveloped also, but you will get confused. So rather becoming confused, say naked. Say naked. Here also we have enveloped and here also we have naked. Here also we have naked. समझ में आई क्या बात है ना? So how many? Learn the rule. Learn the rule. Naked is always 3. Naked is always 3. What is the rule? Naked is always 3. So out of 6, 3 are naked, so obviously 3 are enveloped. Out of 15, 3 are naked, so obviously the remaining 12 are enveloped. Learn the rule. Naked are 3, whether it is DNA or RNA. So my advice to all the people, Hannah, my advice is that please learn the naked. Don't learn the enveloped. Remaining will be enveloped. Hannah, learn the naked. So out of double H A triple P, POP. POP is naked. You can learn a mnemonic. Naked is POP. Naked is POP. You can learn a mnemonic. जो भी बच गया, whatever is left, don't learn that. That will fall in this category. Obviously. So क्या बच गया? HHA. है ना? So don't learn. I'd never learn HHA. So obviously it will fall there. है ना? Because we know total is double H A triple P. ठीक है? So I want you to learn POP. I do not want you to learn HHA. So obviously that will fall there. I want you to learn POP. Say yes. In the same way, out of the total 15, here naked are 3, the mnemonic is PCR. Here the mnemonic is PCR. है ना? The remaining जो भी बच गए, whatever, they all will fall in other. I do not want you to learn that list. I want you to learn POP and PCR. In DNA, the naked is POP. In RNA, the naked is PCR. Say, in DNA, the naked is POP. In RNA, the naked is PCR. Say yes, people. I'm super simplifying it. Say yes, है ना? So DNA में naked क्या है? POP. What is the full form of POP? P ही तो confusing है ना? How many P we have in DNA? You will see, ma'am, in DNA we have 3P. We have 3P. But in POP we are including 2. So which 2 we are including? Which one we are not including? Who is the most senior? सबसे बड़ा कौन है? In DNA who is the senior? Grandfather कौन है इस family का? POX. तो POX grandfather है ना? Give clothes to the grandfather. Give respect to the grandfather. Grandfather है ना? So give clothes. Clothes मतलब envelope. Give envelope to him. So POX is not naked. है ना? It's not good thing to make naked to the grandfather. No, babies naked है चलेगा. The babies can be naked. No, there is no shame in that. Babies can be naked, but don't put grandfather. This is how I learn, so I will never forget that the POX is envelope. है ना? So babies can be naked. Who is the baby? You will say ma'am parvo. Parvo can be naked, है ना? And Pepova is not a baby, लेकिन not grandfather also. So उसको भी naked चलेगा. But pox is never naked. So this is how I learned. A में there is no confusion. It's only adeno. So this is the full form. Parvo, Pepova, and adeno. PAP. Learn it. Howsoever learn, but learn. The same concept I will apply there. Come on the RNA. RNA, we have PCR, but we have two P, na? One is small, one is large. So which P you will include? We have two P. One is baby, one is grandfather. I mean, one is picorna, and one is paramyxo. Which you will include? Obviously, babies can be naked. Apply same concept. Babies can be naked. So I will include parvo. I'm sorry, I will include picorna. I will include picorna. I will not include paramyxo. Paramyxo, I will give the respect. Paramyxo, I will give the respect. I will give the envelope. So I will include here picorna. There are two C, but you have to learn. What are the two C? Kelsey and corona. So it's Kelsey, not corona. ये तो याद करना पड़ेगा. इसको कोई mnemonic नहीं है. There is no mnemonic. And there are three R. So include Rio, not the other two. Not Rhabdo, and not Rota. You have to include Rio virus. ठीक है? Got it? Say, people say. I want all of you to learn. In the DNA, naked is PAP. Learn the full form. Howsoever you can learn. If you like my concept, learn that way. If you don't like my concept, make your own concept. But learn. Ultimate thing you have to learn. है ना? In the RNA, learn naked is PCR. Naked is PCR. But which P, which C, which R? Because we have 2P, we have 2C, we have 3R. So which P? P, though you know, the babies are naked. But C and R, you have to learn. है ना? Got it? Say, people say. Can we proceed? So this is the thing you have to learn, enveloped and non-enveloped. Okay. Can I move this page? Say yes. So this is the same thing written in front of you. So among the DNA, naked is PAP, learn the full form. Among the RNA, naked is PCR, learn the full form. So you can see among the DNA viruses, the 3 are naked. The remaining are enveloped. Among the RNA, the 3 are naked. The remaining 12 are enveloped. I do not want to learn you that 12. Obviously, the remaining will be there. Okay. So read the question, tell me the answer. Please wait, don't jump. I want a RNA virus which is single-stranded and which is non-enveloped. Tell me the answer. Tell me a RNA virus. Tell me a RNA virus which is single-stranded and which is non-enveloped. So RNA virus, reading the word RNA, I will remove POX. POX is a DNA virus. It is not a RNA virus. You know how POX is DNA. वहाँ का grandfather है. Among the remaining three, which is non-enveloped. What do you mean by non-enveloped? What do you mean by non-enveloped? You will say, ma'am, naked. Non-enveloped means they are asking naked. So RNA में naked कौन है? You will say, ma'am, PCR. तो PCR में से कौन आता है? PCR में से. Obviously, Bunia is not there. R is retro. No, this R is Rio, not retro. So retro is not there. But P is Picona. The babies are naked. Learn the mnemonic, babies are naked. Babies can be naked. It's okay. Okay. So you can learn it's Picorna. Say yes, people. Got it? So in this way, you get simple question. The last concept. I'm writing only RNA virus, not DNA. है ना? RNA. So we have 15 RNA. We all know. है ना? So I'm just writing the mnemonic to explain you the last concept here. Okay. So let me tell you. Okay. है ना? Among the RNA, not DNA. Among the RNA, there are 4 viruses among the 15. There are 4 viruses in which RNA is segmented into pieces. RNA is in multiple pieces. I will show you the diagram also. RNA is in pieces. You can learn fragmented, segmented, pieces. एक ही बात है ना? So the mnemonic is BORA. BORA. है ना? You can learn BORA is in pieces. BORA is segmented. है ना? What is B? B is Bunia. है ना? What is O? O is orthomyxo. है ना? What is R? There are 3 R. R is again Rio. हर जगह Rio ही आ रहा है. Only Rio is coming. And A is arena. है ना? So I am taking BORA from the 15 only. From these 15, the RNA is in fragment. Let me show you BORA. What is BORA? They all are RNA. BORA, Bunia, orthomyxo, Rio, arena. BORA. Say yes. Say yes. You have to learn the number of the fragments also. In Bunia, it is 3. In orthomyxo, it's 8. है ना? 8 plus 3, 11. So here it's 11. 10 और 12 के बीच में 11. है ना? 8 plus 3, it's 11. Rio में 11. और arena में 2. This is how I learned. BORA is in pieces. The BORA is in pieces. So 3, है ना? 3, 8. है ना? 11 and 2. Am I right? Yes, I guess I'm right. 3, 8. Because 3 plus 8 is 11. है ना, ऐसा ही था ना, यस यार ऐसा ही था. So bora, BORA, है ना. So here we have 3, 8. 3 plus 8 is 11. 11 मतलब 10 to 12. 11. And here we have 2. This is how you have to learn. Okay. Okay, can we go ahead? That is Bora. Bora is in pieces. So this is a spotter image. Can you identify the virus? There is a big clue given to you in this image. It's a spotter image. Can you identify the virus? You will say, ma'am, I can see many RNA. It's the RNA virus. I can see many RNA. Count how many? Say, ma'am, 1, 2, 3, 4, 5, 6, 7, 8. Oh my God, RNA is in 8 pieces. So what is the virus here? RNA is in 8 pieces. RNA is in 8 pieces. 8 pieces RNA. So obviously my answer is orthomyxo. That is influenza virus. So it's the image of influenza. So you get such concept. As zaruri nahi hai ki they will ask you direct question ki how many pieces orthomyxo. They can give you an image. You apply your concept in the image. In the same way, you can get images of the other viruses. You count the number of pieces and tell me the name of the virus accordingly. Say yes, people. Say yes. Read the question. Tell me the answer. Segmented genome is found in all except segmented genome किसमें नहीं है. What is the mnemonic? The mnemonic is Bora. What is not coming in Bora? Read. What is not coming in Bora? I guess Bunia is coming here. है ना? I guess what is R? Rio. Rio is also coming here. Huh? What about influenza? Influenza is तो orthomyxo है. Orthomyxo is the name of the family. The member is the influenza only. So you should know the members also. है ना? But rhabdo is not coming. So my answer is D. Not A. Sriniket, not A. Some people are saying A. A नहीं होगा. Actually, A is influenza. Influenza is a member of orthomyxo. है ना? Not A and D. Navjot, only D is the answer. Answer is only D. है ना? Not A and D. Because influenza is the other name of orthomyxo. Orthomyxo is the name of the family. Influenza उसका member है. You should know the members also. Okay. Canal Bora. Bora is segmented. I mean to say, Bora. You want the full form or you want the meaning? Here you can see RNA is not continuous. It is in pieces. So there are 4 viruses where the RNA is in pieces. You can count the number of the pieces. There are 4 viruses. The mnemonic is Bora. The mnemonic is Bora. है ना? Bunia, orthomyxo, Rio, and Arena. You have to learn. Orthomyxo is influenza only. That you should know. Okay, you got it, Canal. Now you got it. The RNA is fragmented. Normally, RNA is not in pieces. See, there are 8 pieces. It is not in pieces. It's continuous, but it's not continuous. Normally, RNA should be like this, but it is in 8 pieces. It's not continuous. It's like this. Normally, it's like continuous. Got it? Anna? So only in 4 viruses it is fragmented. Say yes, people. So this is the 4th concept I taught you. Anna, coming on the 5th concept. Till now, what we have studied, you want to revise? You want to revise? If you want to revise, you have to help me in writing the classification. I will ask you 4 questions, you answer me 4 answers. है ना? So DNA, RNA. I'm writing the classification as usual. लिख लिख के बोर हो जाएंगे ये classification आज. But we have to write many times. There is no escape. So that we can learn it, multiple concepts in that. चलो. A, B, C, D or TPH. है ना? You will learn this classification by writing it multiple times. Anyways, the first thing, tell me all DNA viruses are double-stranded. All RNA viruses are single-stranded. We know that. है ना? Tell me a double-stranded DNA virus and tell me a single-stranded RNA virus. पहला concept. Who will tell me the answers? Quick. Tell me the answers. Double-stranded DNA. I'm sorry. I have written opposite, na? All DNA viruses are double-stranded. Tell me a single-stranded DNA virus. All RNA viruses are single-stranded. Tell me a double-stranded RNA virus. That I want to ask. So, of course, the answer here is Parvo and the answer here is Rio. That was the first concept I told you. Please learn that. Okay. The second concept I told you, tell me the smallest here, largest here, smallest here, largest here. So who's the smallest, largest, smallest, largest? I mean the baby and grandfather, the baby and grandfather. Here the baby is Parvo and the grandfather is Pox. Here the baby is Picorna and the grandfather is Paramyxo. And you know overall are DNA only. DNA wale overall bhi hai. So this is the second concept we have seen. See the first concept, see the second concept. The third concept, tell me the naked here out of the 6. Tell me the naked here. Kinke paas kapde nahi hai. They are naked. They are not given the cloth. Babies are usually naked. So here the naked, you will say, ma'am, PAP is naked and here PCR is naked. I assume you know the full form. I'm not saying the full forms. That is the third concept I have given you. The last concept, one more concept I had given you. Tell me the segmented RNA. We don't have segmented DNA. Segmented RNA. 4 segmented RNA, the mnemonic is Bora. So till now we have studied this. Whatever I taught you, I have compiled here. So you know the full forms, I guess. Give me a thumbs up. Can we proceed? We have completed a major chunk here. Got it? Now after this, when I start the viruses one by one, I will teach you 12 viruses. Now it will be fun for you. You know everything about them. क्योंकि maximum चीजें are covered here. It's like the mnemonics of bacteriology. I taught you the mnemonics in bacteriology. Then I taught you the complete bacteriology. It was very easy. You already know this is capsulated because it was coming in the mnemonic. You already know it's forming because it was coming in the mnemonic. In the same way, virology will become super easy for you once you understand the basics. So start with the basic. If I wish, I can skip the basic and I can directly start the virus by virus. It will save my energy, my time, my everything. But understanding wise, it's not good. You will do the rectification. Course will also complete quickly, but it will not good. It is not good for you for retention. Retention wise, this is good. Okay, can we proceed? So these are the 4 concepts we have learned till now. Now let me come on the 5th concept. The 5th concept is the symmetry. The symmetry of the viruses. There are 3 type of symmetry. First, understand the name. These are the 3 type of the symmetry. The first symmetry is icosahedral. The first symmetry is icosahedral. The first symmetry is, what do you mean by icosahedral? Can you see this is an equilateral triangle? This is the equilateral triangle. This is the equilateral. These all are equilateral triangles. Can you see? These all are equilateral triangles. Total 12 equilateral triangles are there. Few are visible, few are behind. And it is looking like a diamond. Total 12 equilateral triangles looking like a diamond. Diamond, it is known as icosahedral symmetry. See the diagram. This is known as icosahedral. This is known as helical. What is helical? It is a spring-like. Have you seen the spring? Spring देखा है? हाँ? Spring. It's like a spring. ठीक है? And complex, no fixed shape. So total 3 symmetries are there. Say the name. Say, ma'am, one is icosahedral, one is helical, one is complex. Learn the name. Now tell me there are 6 DNA and there are 15 RNA. ठीक है? Among them, which one is having these symmetries and which one is having these symmetries? ठीक है? Viruses wise we will see. ठीक है? Let's start with DNA. Let's start with DNA. These are the DNA, these are the RNA. Can you see this classification? There are 6 DNA. See the 6 DNA. सब लोग देखो screen पे. The 6 DNA. All DNA viruses except the grandfather. Except the grandfather. Grandfather is pox. Remaining 5. They are icosahedral. They are diamonds. They are diamonds. है ना? But the pox is the grandfather. उसने बोला मैं तो complex बनूंगा. I'm complex. I'm the grandfather. So the grandfather, you can learn the grandfather, the pox is coming here, and the remaining all DNA, the remaining all 5 DNA are coming here. Say yes. So can I say none of the DNA is helical? Yes. So read the 3 statement. All DNA are icosahedral. Say yes. Pox is complex and none of the DNA is helical. You understood these 3 lines? If you understood, say yes. This is regarding the DNA. This is regarding DNA. The summary is written in front of you. Pox is complex because he is senior. He say कि मैं usual नहीं लूँगा. You all are taking diamond, it's okay. But I am senior, I should get some privilege. So I'm taking the complex symmetry. ठीक है, you learn like that. And remaining all are diamonds. है ना? They are icosahedral. है ना? None of them is helical. No. So you learn the story. Helical को बुरा लग गया. Helical is feeling bad. Am I too bad? None of the DNA viruses allotted to me. So we said it's okay. Don't feel bad. The major chunk of the RNA we will give it to you. I mean the all 12 enveloped RNA we have given the helical because the helical was feeling bad. None of the DNA is given to him. It's like a story I'm helping you to learn. Helical को बुरा लग गया तो उसको RNA का major chunk दे दो. 12 virus. The enveloped viruses are helical and the non-enveloped are again diamonds. That is icosahedral. None of the RNA is complex. Got my point? All enveloped are helical. They are 12 in number. All naked are icosahedral. They are 3 in number. And none of the RNA is complex. Say yes, people. I tried very hard to explain. So in this classification, you can understand. Anna? So you can say all DNA except pox. I mean the 5 DNA. Anna? And pox is separate. So all DNA are icosahedral, but pox is complex. Anna? This is all about the DNA viruses. Among the RNA, say enveloped and naked. Enveloped and naked. Enveloped are 12, naked are 3. You tell me the enveloped one are helical and the naked are icosahedral. This is the summary. You can write like this. So you get questions on that sometime. That's symmetry. Read the question, tell me the answer. Which of the following virus do not have icosahedral? Do not have icosahedral. Read. Parvo, Pepova, Pox or Adeno. They all are DNA. Happy में आते हैं सब. Double H A triple P. See, they all are DNA. So among the DNA which is not icosahedral, only one is not icosahedral, Pox. Because Pox is complex. Remaining all are icosahedral. So very frequently asked PYQ. Pox is complex. है ना? Coming on the last thing, the shape of the viruses. Coming on the last thing, the shape of the viruses. है ना? Please learn the important shape. I will teach you virus wise also. You can see pox virus is brick shape. Can you see it here in the diagram also? It's brick. It's looking like a brick. ईंट के जैसा है. Please learn rabies virus is bullet shape. बंदूक की गोली. It's like bullet. Can you see? It's like bullet. It's like bullet shape. है ना? And the adenovirus is space vehicle shape. Can you see it here? It's space vehicle. Also known as satellite. Satellite. You can learn both. एक ही बात है. Space vehicle or satellite. Say space vehicle or satellite shape. Say yes, these are the shapes. Please learn. Learn the important three shapes on which all three PYQs, image-based questions also come. Image-based question already asked on this. है ना? Space vehicle पे आ चुका है. Say yes, people say yes, respond. Got it? Say yes or say no. Got it? Any confusion? Anyone has any confusion till now? Say something. Am I live? Okay. Chalo. Which of the following virus is bullet shape? Which of the following virus is bullet shape? Yes, of course, rabies. I know. And if I change, it is brick shape. Answer will become pox. And if I change satellite shape, option is not there, adeno will be there. So you get questions on that. Got my point? Got my point? Any confusion till now? Okay. So after that, one more thing I have to teach you: replication of the viruses. Replication and inclusion body. दो चीजें और पढ़ानी है. Then I will start the 12 viruses one by one. So we will require 10 minutes on each virus, 5 or 10 minutes. Some viruses are small, some are large. So 10 or 15 minutes on each virus. So till the evening, we will complete all 12. Before that, I have to finish replication. Replication of the virus and inclusion body. And then we will start virus by virus. So you already know which is double-stranded virus, which is single-stranded virus. You already know which is enveloped virus, which is naked virus. You already know which is smallest, which is largest. You already know which of them is segmented. You already know the symmetry of all the viruses. And you already know the last thing, the shape of the viruses, the important shapes. Okay, so these things we have already completed in general. So can we take a break now? After that, we will start virology in detail, one by one. I guess I will be able to finish entire virology in 3 hours. Maybe 3 or 3.5 hours. So whatever time left now, in which we can complete either mycology today only, or we can complete the remaining immunology, whatever. So virology is a small section. We will complete till evening. Okay. So let's take a break. So currently it's 12:20. So let's take a break till 1 o'clock. Can I come back at 1? At 1? Maybe at 1:05. Okay, so this is your lunch break finally. I'm not going to give any other lunch break. So this is your final lunch break. After that, we will continue. Maybe we will take a coffee break at 4 o'clock or something. Okay, whatever, whenever I will feel down. So this is your lunch break. So let's take a little bit larger, 1:10. I will have my lunch also, breakfast, lunch, whatever you want to take. So this is your lunch break. Okay, so let's meet again at 1:10. Okay, ji? So break till 1:10. Okay. So I'm ending the session. You will get a new link.